Turn on auto. So let you quit. Battling. Oh, are you doing it too? I am, yes.
Good morning, everyone. Welcome to Dive 537. Back around Dorado Outcrop. We have Emily Cowell from Temple University. Is the scientist in charge of the dive, and she'll be doing the narration. Pilot is Chris, and co-pilot is Adam today.
Hi everyone, so we've just touched down on Dorado Outcrop. We're already seeing some octopus that look like they're in that upside down kind of brooding position. But our priority, just to start with, is we heading up to marker W that was put down from previous dives. That's a spot that we've done some sampling at before. So I think we're just heading slightly upslope towards that. Then once we get there, the main objectives of today are deploying some kit. So we're deploying Osmo samplers and temperature probes. We'll be taking some temperature measurements as well as some water samples. We're doing that at a couple of sites across the top of Dorado Outcrop. And then other than that, the main target of today's dive will actually be video. Getting really nice, clear video of behaviors of the octopus, possibly doing some biology sampling as well. Looks like you can see some weights from previous subs.
Oh.
Some of the subs work by dropping their weights before they come back up to the surface. So those kind of weight plates there, those square things are what you're seeing. Some beautiful fan corals. Just getting all of the settings on the cameras adjusted as well.
Well, the problem is I don't want this taking off half the screen.
Great. Yeah, so if we can head up to that waypoint, then there should be a marker there that we can find that should say marker W. I know, there's so much amazing stuff here. It's gorgeous. This C pen kind of thing? Yeah?
Not that one, but the one on the right.
I don't have the pointed stick. This thing at the bottom, right here, I think. Is that the one you're looking? I get a video of it. Yeah. Can I put the pointed stick? You just want to see a really clear video. Yeah, if we could just get a really nice zoomed in video of the polyps of that would be great. You're not working the sample then? Just like a good
Can you come down
Sounds
just
good.
a little bit on that?
Is this something like before you wanted to put a video of it? I don't know if I want to collect it, but
And focus.
yeah. Sounds good. Cool. Do you need the lasers on as well? Yeah.
Go and come back to that. Just to be too close.
Okay. Yeah, if we can just get a really good close-up view of the polyps, I guess, while we're this close.
Yeah.
Oh, no, it's gone too far the other way, I think, maybe. Around there. Oh,
Very sensitive, so it might be moving it right down to any little variation in the lighting and color. So.
okay. That's enough? You think? You don't want the lasers on and things as well?
Oh, sorry, lasers on?
They could put them on quickly.
Yeah,
if you wanted.
lasers are on.
Oh, there it is. Did you get a clear enough view of the polyps there? No, it's really fine. Okay. Cool. Okay. And in that case, I think that was enough. So
Okay.
we could probably keep heading up to marker W, please.
Alright. So, that's awesome to know.
Thank you.
Try the auto focus. Roger. Are the lasers off? Lasers are off.
Yeah, there you go.
No, it's the Fort Ramp. Oh yeah, that's nice to have out there. Nothing to see coming up in front of us. Nothing to see. Really bright. Sure.
You're seeing such a really amazing variety of things on the top of this outcrop. It's a really beautiful sight. It's noticeable as well the much higher presence of these really big fan corals compared to what we were seeing on the sides of the seamounts, where it's obviously more unstable.
Whoa, hey, buddy.
Oh. Got a visitor. Oop. It should actually be a physical marker.
Yeah,
That
that
should-
should be it, it's right there.
Is that
That's marker
Lewis?
K, and marker
Lewis?
W is just past it.
Yeah.
Oh yeah, I see. So you can see one of the markers just at the bottom of the screen there. Marker K, which is a spot where we'll be putting some more of our kit in a second. And then this down here.
You turn what was that? An R?
That one says R. Oh, W here on the right.
Is that-
Pan right slightly. There we go.
Okay, and that's where we're on at W?
Yeah, W is the spot. Thank you.
Couple,
Hmm.
couple octopus.
Yeah, just a couple. It's going to be hard to find any.
Jump seat. This.
That's,
I think that's about it. Hello. Oh, yeah.
just grab them. Yeah, we're gonna leave them so the big put eggs in them.
I'm gonna try to find a spot to set it down without disrupting any of these guys.
Yeah, I think we've generally him sitting in that bare spot right underneath the marker W.
Good afternoon, good evening, good morning. This is Jorge Cortez from University of Costa Rica, and we're here back on the Octopus Odyssey Expedition aboard the Falcor 2 off the Pacific coast of Costa Rica. And we're back to Dorado Outcrop, where we visited about a week ago. That was our first study site. And looking at the deployments we left, see if everything is in order, and we're gonna be doing some sampling here and collections. Buenas tardes, buenas noches, buenos días. Les habla Jorge Cortés de la Universidad de Costa Rica. Estamos aquí de vuelta al afloramiento llamado Dorado. Aquí estuvimos hace una semana y ahí vemos los experimentos que hemos dejado, revisando que esté todo bien, y vamos a retomar algunos de los estudios que empezamos hace una semana, hacer unos videos y muestras adicionales. Remember to send in questions, comments. Acuérdense de enviar saludos, comentarios, preguntas. Aquí con mucho gusto colaboramos, contestamos lo que podamos. Sí, aquí vemos una gran cantidad de plumas de mar. Son esos, se ven como una pluma con un colorcito como anaranjado, tapizan las rocas en esta zona. Y vemos los famosos pulpos, que es lo que hemos venido a estudiar. Yeah, we see a lot of this sea pan, orange color, attached to the rocks, and the octopus that we came originally here for.
That was the loudest whisper I've ever heard.
With me here is pilot Chris and Adam, and we'll be changing pilots pretty soon. And on the transmission there is Alex. Hello.
I'm here, Chris. So.
You want to sit?
Sorry. Yeah, maybe that would be helpful. Thank you. Do you want your phone and your book? Oh, so sorry. Hi.
Hi. Fancy seeing you here.
Mm-hmm. Okay, so our first order of operations is to look for some venting. So I'm going to do a little bit of zooming. That's okay? You're all stable there?
All stable.
Why is the autofocus not working? What do we need to do to make that work?
I don't know.
That's better. Yeah, thank you.
There you go.
Thank you. I feel like the shimmering might not be coming from where they are, but from down below.
Okay, well, it's hard to tell with just the video because I think they're shimmering in front of shimmering. So if we could get out the high T probe, and we're gonna try to query some of these holes like here and down here to see if we can find a good spot for putting some of our experiments.
Okay. Want to draw out for you a little bit? How's that?
That's good.
Okay.
Thanks to our audience for joining the Octopus Odyssey Expedition. Today's dive is visiting the Dorado Outcrop offshore Costa Rica. This is an area with known low temperature hydrothermal venting. Maybe you can see in the video a little bit of shimmering water around some of these octopus. These waters are warm to about 12 degrees Celsius as compared to background seawater, which is more like 2 degrees Celsius. And we're returning to the site today to put out some experiments that we'll be recovering back in December. Right now we're setting up to measure the temperature of the water to figure out where some good spots are for putting our experiments. Which one do you want to start with, Ben? You want to start with that big one? Okay. I can. And can we get the temperature readout on the overlay?
Oh, yeah.
Thank you. So now you can see the shimmering water quite a bit better. But it might be that the shimmering is coming from a vent just below us and not actually coming from that little spot. A little bit further so that it's really in the whole area. If you don't mind. And if the manipulator allows, yeah. Let's see if there's any indication of warmth back there. So far, not really. Yeah, okay. So deceiving little spot. We can go ahead and pull the temperature probe out, Ben. Well, let's see where we want to go next. Maybe down here. Let me just zoom in real quick. Yeah, maybe this spot right here above the shrimp, if you can. Janet, do you think that's a male?
I suspect it is, but I can't say for sure.
Yeah, okay. Okay. Well, the temperature's a little bit warmed, but it still looks like a lot of mixture with bottom seawater at this spot. Okay, we can come back out, Ben. Yeah, is the porch out?
Not all the way.
Can we bring it in a little bit so that I can see down below us a little better?
You ready then?
Yeah.
Okay, cool.
That hole underneath.
Yeah, I was trying to avoid having to go down there since there's so much other stuff in the way. But we may need to reposition to be down there since this site isn't quite as warm.
That hole right underneath.
What are you pointing at, Ben?
That one, just, this one?
Yeah, I think all the fluid we're seeing is actually coming from here and just coming up this wall. So we might need to reposition to get down in this area to put our stuff out.
Right.
So Scott, you might need to fly down there.
That,
Are we gonna do more temperature probes?
Yeah.
When we get there?
Yeah.
Just keep it in your
Yep.
loft.
Yeah, so there's like another little bare spot just to the
Just
starboard
underneath.
side of us.
Just to the right of this marker. Yeah.
Exactly, yeah.
okay, that's good.
Great. Okay.
Yeah, just in that spot there.
If that rock slips.
Okay, is he gonna be able to reach up to that pot?
Can you reach the clay pot? You think then?
Yeah.
Okay, so then, let me just have a look up here.
I see. You wanna try over there?
Yeah, I'm just trying to think about where we would put the experiment once we do the measurements. Because there's just so much other stuff here. Including Octopus, who want to be here also. Yeah, do you think you can reach over here or is that too far away?
Okay.
Okay. Yep.
Settle down.
I'm gonna give it a little bit of push.
Hold on. Yeah. Can you point that again, Ben?
Yes, this
Okay.
right over there. Let me know when you want me to zoom.
Coming up. There's a good landing spot over there if I need to reposition.
Where? Over there.
Right next to the W.
Yeah.
I can put the
I think that's where we just were. Right, because you see marker K right there. We were just sitting right here.
Yeah, I was gonna go like
Ah, okay.
kind of like 8 o'clock.
No, you know what? It won't matter with this one.
Yeah, let me reposition.
It's like rubbing off calluses. I'm gonna just hold this up.
Ready?
Yeah. 3,
This is quite difficult terrain to navigate in. It's very steep here, lots of loose rocks.
What's going on right now is your auto heading. Okay. So see how far off your set point is from where you're at? It's just going to start throwing a tornado. Okay. You need to try to get in a stable position to come up with it. So right now you're facing kind of towards the vessel right now. Your locals are going to the stone, right? So just keep that in mind when you're laying down. Probably put a little bit of flat land here. And that will counter what your auto heading is going to do. So come down, settle it, just a little bit of lateral point, and then you can lock that up.
Let me know if you want me to pan this down or anything.
It was right there, right? What we're looking for?
Well, sort of, like anywhere in here. Yeah, so we want to be able to deploy stuff right in this area, and we're trying to do some temperature measurements over there. She just left.
She did commenting on the non-breeding captured octopus. Instead of looking at everyone we can go by, it seems like they're, you know, I don't know.
She said she was looking at it and couldn't tell the sex.
Yeah, I've been watching really closely when I get the chance to see those arms, and
Yep.
I mean, a few of definitely. So I think it's just. So yeah, not that aggressively.
Okay. So in the position we're at now, is it okay if I move the camera, Ben? Okay, let me just assess again what I want to try to do. I think ideally, we probably do still want to get our measurements and stuff in right where this pot is. Janet, is it okay if I temporarily move your pot?
Sure.
So that we can try to get some temperature and squeezer samples right there?
Right, I think that's right where Jeff's Osmo sampler hit.
Yeah,
The
exactly.
hotspot.
Yeah. So I know you've got the temperature probe in your manip. I don't know if we can maybe use the port manip to just grab this clay pot and like move it out of the way for a moment.
It's very, looks like it's like at all the ones that are breeding, and I think I've seen a handful of females. A handful of females? Yeah. I haven't seen one yet, but I mean, there's some that I can't tell, but I've just been sitting up there just staring at it like,
The question is what, yeah, I think that telling all the lanyards from each other is a little tricky. You can probably just grab that pot if you don't. So I,
Orange. Using a clay pot.
yeah, I think that orange lanyard. It's also okay if you pick up one of the other ones. One of the interesting things about our return to Dorado Outcrop this year, as compared to 2013 and 2014, where dives were last done here, is that there's even more octopus here than the last time. Yeah. And do you need me to move somewhere?
Yeah, just look at that. I think what I'm gonna do is I'm gonna drop the probe here, starboard arm for a moment.
Okay.
See a question of what's in the pot? Nothing currently. However, that was left here along with that chunk of pipe on the right there, as kind of possibly targets for the octopus to inhabit or brood at.
Sorry, Ben, please don't stick stuff in that milk crate. It's fine to go in the other one, just not this one with all the green and yellow, because there's some cables in there that we don't want to get.
Can I just put it on top?
Yeah, you can put it on top for sure.
Sorry.
It's okay. Okay.
So it's only the orange handle, right?
I think
Yeah.
so. So you may notice in the frame here that many of these octopus are sitting in a relatively unique position. Their arms are coming up around their mantle and potentially protecting something behind them, although we're discussing here if all of these octopus are in the brooding position or maybe in a different position or doing something else. But some of these are likely females that are brooding their eggs and they're protecting their eggs in that position.
Can you bring the ram in just a minute?
Touch more?
Yeah, touch more.
Just scooting a little closer to the cliff here.
Great. And if we can maybe temporarily move it over to our right, over by this other PVC shelter, and then we'll move it once we're done.
Point it again.
That was right here.
Yeah, just kind of in this area. Doesn't need to go somewhere in particular. I just want to try to get it out of our way.
You have here.
Yeah, I think that's fine. Okay. So we can again grab the temperature probe. Yeah, we just want to be really careful. See, at the tip of the temperature probe is right next to some tubing. Okay, so where we move that clay pot, kind of behind this octopus here, is I think where the majority of the flow is coming out. So we want to try to get back in that area. It's behind the octopus.
Can you zoom in there?
Yep. Yeah, so, oops, too close. Good thing it has a soft point.
Yep.
So as we're setting up to do this temperature measurement, you might be wondering what some of this other stuff is on the seafloor that we've put down here. So these are different scientific experiments. Can we get the temperature overlay on again, please, Alex? All right. So the experiment on the left-hand side is what we're calling a microbe trap or a microbe colonization experiment for studying microbes that live on rocks. There's different types of sterilized rocks inside those little PVC tubes. Yeah, I think we want to try to get a little bit lower and behind that octopus if possible. Yeah. Yeah, we just want to be like behind the octopus, whether it's from above or below. but I think that's where the hot water is coming from. This is a really unique adaptation that these octopus seem to have, that they preferentially come into these warmer waters. This is quite a big temperature difference compared to bottom seawater. Is it possible to go any further back then, or do you feel like you're okay? Okay, so I think the max we've registered so far is 8.79, 8. Oh, there we're going a little bit, 9.2. We've previously measured temperatures around 12 degrees C here. Okay, no, 9.67. The shrimp over on the right?
No, just about the octopus where I am right
Oh,
now. I just wasn't sure about the rules of engagement.
fair enough.
That's the animal. But I had that explanation.
Not quite as warm, but I mean, this is fine. I think where we were just a moment ago is probably where we'll try to get our sample. Yeah. It's very subtle differences, just a few centimeters away. Okay, Ben, I think we can go ahead and put the temperature probe back. Thanks for that temperature overlay, Alex. I think we can pull that off.
Are we gonna use this again?
Not right now. We're going to do some other operations.
But we're still gonna use this, right?
When we get to our next site.
So one, back the whole-
Probably, yeah, it's better to holster it. Because the next thing we're going to do is try to fire some of these squeezers.
Can you zoom in to the set?
So back to what I was explaining before, the device on the left-hand side with the yellow weight on the bottom, those were microbe colonization experiments. You see that there's several of them deployed in this area. We put them down a few days ago. Each one of those little PVC chambers has a different type of rock in it. That our goal is to see what microbes colonize these rocks and how they compare to the microbes that are on the rocks that we collect from this environment. You also see some larger PVC pipe segments. Those are animal shelters. The idea being that some of the octopus might go inside of them and maybe lay their eggs. So when we come back in December, that will be a way that we can try to collect octopus eggs without having to try to scrape them off of the rocks. Okay, so in this forward starboard basket, there's 4 squeezers. If we can start with the one that's right here, outboard starboard.
I'm gonna put this up spot, and then I'll pass it on to the
That red handle. All right, this is number 5. This instrument we call a squeezer. Behind the white plastic is a trigger that allows a spring to pull a pin back, pulling the plungers up on the bottom piece. That's a glass cylinder, and that allows us to collect a water sample. The idea behind these is that they can be a one manipulator operation once the manipulator has it in his hand. Let me know where you're ready for Zoom, Scott.
Got my arms slowed way down, so. Taking my time. And that little movement after freeze is annoying. Okay, you start zooming back.
Might be an awkward shot, but
Yeah, it's sitting there, it seems that's good.
can you get some highlights as we're going in, just so that we can try to see? Yeah.
Maybe you could zoom in at the bottom. There you go.
How close do you want me to get?
I think, yeah, this is probably, I would hold here. Let's let it settle for a second.
Yeah.
Okay, let's try to fire. Okay. Before you move, hold on one second so we get a highlight. Okay, and then this is squeezer 5. Great. We can go ahead and try to stow that. And can you comment that like it's right on an octopus? That way we know for the microbiome sampling.
You're not wrong.
Can you zoom out?
Yes.
You want to grab it? No, put it in.
I think let's try and drop it a little bit.
Okay. Okay.
There you go.
We're getting requests for narrating and staff. Would you mind?
Buenas tardes, aquí Jorge Cortés de la Universidad de Costa Rica. Lo que hemos estado haciendo ahora, hemos vuelto al sitio Dorado, donde estuvimos hace una semana, empezamos esta expedición, ya que están el sitio de los pulpos, y lo que se ha hecho son mediciones de temperatura, a donde están saliendo esas aguas un poco más calientes y encontramos hasta casi 11 grados centígrados. Y ahora con ese instrumento que están viendo ahí, están guardando, eso es como una jeringa gigante y entonces eso se acerca a donde están saliendo los fluidos y se toma una muestra. Entonces ahí va sellado una muestra de agua de ahí, y eso va a permitir hacer toda una serie de estudios sobre la química y microbiología de esa agua. Y vamos a continuar.
Do you guys want me to tell you my next operation, just in case it affects your planning? So the next thing we want to do is this large milk crate here. We want to unbungee it, and then we want to put it on the rocks in front of us. On the outboard side, well, actually, right on top is all the cabling. The ROV float is holding the end of it, which is what we want to, that's the business end that we want to get into the octopus. So I don't know if you want to put the milk crate down and then grab the ROV. So there's this red loop is the bridle for the milk crate.
Are we leaving the milk crate?
We are leaving the milk crate. Yeah, so the bridle here, you want to pick it up, put it down, and then we'll grab the ROV float and like try to pull that cabling off and then put it behind that octopus.
It's this one. Okay. I'll do this right away. Yeah, there's a monkey fish on the bungee.
Yeah, it's right here.
Of course, it's underneath.
Yeah, of course it's underneath. Yeah, of course.
Was there a knot tying night? There we go. For the scientists, or was this mostly one person cranking away at them?
The scientist didn't make this knot. This was a SuBastian team knot. I say anything about the positioning of the knot. I'm just saying making the knot.
Yeah.
We're all part of the same team here.
That's it. It's just a little bit of some metal right there. So we'll just pull it that way.
You want to zoom on the center?
If you can zoom in on the science camera.
Whoops.
And just following it. Of course, it's underneath the.
Yeah. So it's a little.
Yeah.
Right there. See ya. Yeah, no, the reason I'd asked is I know that the first time I'd ever tied a monkey's fist or had to learn, there were some scientists that just needed them made, and they came and walked us
Alright.
through it and left us a bunch of rope.
Okay. I don't know how to make these knots, so maybe I should take that class.
I'll be more than happy to give you a class after this.
Okay.
They're fun.
And this whole thing weighs about 8 pounds, just so you know.
I think no harm can handle that.
Okay.
See that?
Yeah.
Okay, we're going to go. We want to put this other side.
Kind of right in front of us,
All right.
like on these rocks, wherever.
But then I'll put this. I guess we'll put it away. Is there anything underneath?
I don't think there's anything right underneath
No,
you.
right there's all right.
Happy?
Yeah, that would be a great spot.
There we go.
Still works. Okay, so now the little W wheel is just gently rubber banded on the edge of that milk crate, and as you pull it, you'll want to pull up, like out of the milk crate, because at the bottom of it is another weight with the intake for this tubing,
Right.
and it's currently stuck in a little holster. So we just need to pull it out, and then all the other tubing will come with it. Hopefully. Yep, it's coming.
Cool.
Awesome. And so we want to try to stick that white tube behind that octopus as much as we can. What's that?
Feather underneath?
Yeah, this, I'm hoping that it's just sitting on top. But yeah, there's about like 12 feet of cabling here with that tubing in
Make
it.
sure that I didn't drag it.
Nope, you're good. There should be plenty of play there. Assuming that the rubber bands come undone.
Yeah, like packing a parachute.
I would never.
No, this
yeah,
is
in
cool.
In a way, yeah.
I like it. Yeah, it's nice modularity.
If it's not easy to position by holding the ROV float, it's also fine to just grab that dive weight and kind of push it in if you think that's easier.
Is it this octopus, right?
Yeah, the one right behind the weight, right back in there.
Can you point it?
Oops. We want to try to get it right back in there. So this instrument that we're deploying is called an Osmo sampler. What it does is continuously collects about 0.5 milliliter of fluid, about 1/2 teaspoon of fluid every day, continuously. It doesn't require any power or batteries. Oh, are we sliding backwards? Yep. Okay. Good.
I'm worried where it's sitting on the basket now. Might have to pick the weight up.
Okay. I'm going to zoom back in for you, Ben, as much as I can with this bridle in the way. Luckily, the way the basket's positioned, there's nothing important on the backside.
Okay.
Yeah, the way I can see it there, we're fine.
Yeah.
Just hopefully it doesn't roll down the hill.
Yeah. I'm worried if when I lift off.
Yeah, we'll see what happens.
I'm gonna pull a little bit more out.
Yeah, I think that's a good idea. Just gonna look down while you're doing that. Yeah, unfortunately, where the rubber bands hasn't broken yet, so it's partially holding you in place.
Can you look up for a moment?
Yep.
Try not to make any of this back. There you go.
So what you see here, the little white tube is protecting the intake for this fluid sampler. Dive weight is just to hold it down in place so it doesn't float away. The yellow rope allows us to kind of move this about without having to put weight on the actual tubing itself, which is protected inside that clear plastic tubing. It's very small tubing on the inside there.
Fine. Are you happy with that one?
For right now, I am. We're going to put one more other thing on here, so it might get juggled anyway. But that looks good for now. Can you get a highlight, please, Myla? Okay. So now, thank you very much, Ben. Yeah, we're sliding again.
Hold on.
Yeah.
Hold on. Okay. Let's bring that basket on. Hey, Ben.
You see that
Let me
at the end of that tube
Okay.
is an intake for the
It's on
ocean.
there.
Yeah. It's both.
We're wobbling. It's right next to my miles steel.
Happy?
Well, we can move it.
Yeah, we're gonna have to, because that one's gonna leach for sure. It's gonna leach to
No.
his iron.
Okay.
So and definitely don't want your intake right next to where I
Okay.
where the time of the iron that we leach for the next maybe month and a half.
Okay.
Is that why?
Yeah. I mean, the intake is closer to the venting than your stuff is, so it's just going to be blowing it out.
Just based on the flux paper, it seems that it gets pretty intense around that substrate.
Mhm.
It's your call. I mean,
Yep.
You know how crazy it got in the flux.
Yep. You may not want to put it too far up just because we're going to have to go around it again to put some more things down. Yeah.
Oh, that's final note on that. You're on the right, right? Right here. That looks pretty good right there.
We're,
That's red, that's not orange. Perfect. So never mind. That's 23 million euro basalt. That's local basalt. That's on
Okay.
that one.
yeah, your orange one's over here.
My orange is over there. Freaked out at first.
Yeah. It's always good to check. You set, Ben? Okay. The next thing we want to do is grab one of these temperature probes that's in BioBox 1B. Doesn't matter which one.
We're gonna have to pork chop. Maybe not.
I'm not sure which one's on top. Looks like this skate wheel, the one on the left, is holding the one that's on the top.
But you want the one on the left, right?
Yeah, let's try that one first just because I think it'll come out easier. But what do I know?
This one.
Yeah. And that's temperature probe A. And this goes under deployments.
How are they?
Oh, the tape is probably stuck together. So maybe just jangle them around to try to get them free from each other. That
sticky tape.
it's sticky tape. Yeah, we'll try to use electrical tape next time. Otherwise, we're gonna have to pull them apart with 2 minutes.
Or drag it across the edge of the box. Okay, you want me to grab the other one?
Yeah. Here you go. Hang on.
Got it?
For the Osmo sample?
It didn't like our threat.
No.
Okay, so now we have A. Great. So again, we want to kind of try to get that business end at the bottom of the T where that intake is that we just deployed. And if you need to do it by picking up the T handle, that's fine.
Can you zoom in on still?
So we need to log the Osmo sampler as a sample. No, sorry, no,
What's that?
just a deployment. Okay, and that's also just a deployment.
That will be recording temperature.
Yeah, it's been logging since it left California.
Okay.
I think it's set to record every minute, maybe. And when we come back in December, we'll recover it, and Andy will download the data. He probably has to send it to a company to download it, but that's the idea. So it's an autonomous temperature logger inside of this T handle.
Okay. Let me know when you're. I'm gonna zoom out.
Maybe.
Okay.
This one down.
Okay, I'm gonna try to just bump a foot forward here. Actually, hold on a second.
Can we just pull in on the ram?
Yeah, try to.
Not gonna tell you how to drive.
No, that's, I'm new here. Looks like the front porch is getting, so what if I porch it? And then ram.
Yes. Please, please, please.
Aquí continuamos poniendo experimentos. Este instrumento que está en esa caja negra es un Osmo regulador. Eso está tomando muestras periódicamente y analizándolas y lo que estamos tratando ahora es de poner un termómetro, que lo ven ahí en esa T, y eso va a estar registrando temperaturas continuamente. Y en diciembre, cuando volvamos, recogemos todos esos experimentos y se hace toda una serie de análisis para entender cómo funcionan estas ventanas termales. Y ese es uno de los objetivos de esta expedición, es entender estos sistemas que, como se ha mencionado, son muy poco comunes. Y la presencia de los pulpos aquí es todavía menos común. Y es lo que hace interesante, tan interesante y valioso este sitio.
Thank you.
Can you zoom in on the science camera
Yep.
and the tip? Are you happy there, Ben?
Yeah, I mean, let's see how it lands. We might have to push it forward once the T is actually down. But in principle, yes.
Holding on to it. Just go and shove it in.
Again, Ben?
I'm going to let it go and then I'll go push it in.
I think that's a good idea.
Can you zoom in on it? Yeah.
I'll do my best.
You're about 6 inches away from it.
What's on top of my... No, thank you. It's hard to tell. Yeah, it looks like there's a rock. So if you want to maybe pause for a second, Ben. In the sit cam, you can see that the intake and the T-probe are kind of going like this. If we think it's possible to put the T-probe where the intake is, that would be preferred. But if it gets too annoying, we'll just leave it like this.
Can you zoom out a little bit?
Yeah. I think that's good. Can you take a highlight picture, please? Yeah, let's see if it stays there when you let go.
No, this one's years. Let me do it again.
Okay.
Can you zoom in a little bit on the sit?
Zoom in.
Yeah, on the sit.
Where's Betty?
It was just Specimen. Yeah, it's there.
Okay, that's probably good then. Okay. Can you take a highlight, please, Milo? Okay, and then to Janet, would it work for you for us to relocate the pot basically sitting on top of the T-probe handle? Okay. All right.
All good?
Got a shift change going on, so people be patient for a moment.
You turn the camera on in a minute. Are we going to reposition this a little bit to fit onto that perch?
No. Stay where you are. We're going to pick up this pot and put it back where it was, basically on top of this handle.
Okay.
So we're just trying to put all the things right there.
Copy that. One more time. See this pop right here? That pop right there is going just sitting on top of this little T handle. On top of the T handle?
Yeah. And to the degree possible to not like totally disturb the T handle, but we'll see what happens.
And what's the task after this?
We're just going to get a little bit of targeted video, and then we're going to hop up and go. Where are we going? To marker R, which is just around the corner, like It's off to our port side.
You said a marker?
Yeah. When we back away from this wall, you'll see that there's another marker off to our port side. Not that one.
Okay.
We're at marker W, there's a marker K right above us, and there's a marker R just over to the side.
Wow, those tapes up there are really cool.
Mm-hmm. Yeah, we might just look at them after we're done with this.
So do you want them to go a little bit further back or keep coming down where he's at?
A little bit further back and then down, if possible.
Yep.
How's this looking?
Just shoulder right, just a touch. There you go. Now start coming down from to your left touch. That way you're not down on that one monkey. Come up a little bit. Come to your left touch. Alright, start coming down nice and easy like that. What do you think about that?
That looks good.
Just slowly let it settle. If it does anything by it, just be ready to pump it on. So go slack. Slow it in. If you can, pitch out just a touch, so make sure you're slack. Yeah, so you still have a little tension on it. Is it okay if it rocks back that way?
Yep.
Just keep coming down with it, a little bit more pitch out. All right, you're pretty much settled in there.
You get a highlight picture, please.
Do you think we could nudge Gus's experiment just like an inch this way so this female could get back on her clutch?
Potentially.
Yep, all right, so you want us to move that one back just a touch, correct?
Yeah, I'm trying to figure out where so that it actually gets flow. So yeah, if we can move it towards the T handle.
Just a little bit.
Yeah, so you see how this, the yellow weight and the two pieces of PVC are just like really close to the eggs and the octopus can't get back onto it. So if we can flip that up and just go a little bit to slower, yeah, and give her enough room to get back onto it. Good. So we can really grab it, try to grab and bring it, yeah, kind of like that one towards your arm, and then go back to that other direction. Just like slide it over.
It might. Would it be easier to just grab the PVC at the bottom and push it over as opposed to getting it on the rope?
I don't think so. 'Cause
Okay.
The jaws, you could potentially, when you go to grab it, rotate it and
Oh.
hit those eggs pretty hard.
Okay.
When he grabs rope, you can slide
Okay.
it back and then move it into position.
Yeah. Thank you.
Now just go to the starboard. Now start coming down nice and easy right there. See if it settles out.
Thank you.
There you go. Go ahead and look up. That good, Jen?
She happy, Mama.
Yes.
All right, so get right back on there.
You love it.
All right. You want to have a look at those eggs when he just arm back?
Yeah, let's go ahead and pull the arm back.
You get to do the rest of this thing?
Yeah.
So if before we get to eggs, if we could, this one that's right down here at the bottom of the frame that has its arms moving, if we could just kind of get a close up in of that and hold it. That's good. Okay, we can come wide.
I think that one that was had its eggs exposed up there has to move back onto
Yep.
its eggs.
Yeah, and it's not a good shot anyway with the rope in the way.
Yeah. And I could zoom past it or focus past it, but she's really covered up her eggs now.
Yeah, I don't think that's a good shot.
You can have the camera if you want to have a look around here.
Okay. Janet, is there a particular octopus you'd like to spend any time looking at?
Hoping the big ones, I think the males turn just a little bit so I can confirm that.
Which ones are those?
The one just above dead center. Remember sexual size dimorphism? The bigger one there and there.
Okay.
That one too, maybe. I think he is. That's his fourth right arm. We're seeing right at the shrimp tail.
Mm-hmm.
He's protecting it.
You said he's protecting it?
Yeah, his copulatory organ. Males do that.
I've seen a lot of similar scarring on the octopus.
Yeah. Like little heart-shaped scars.
Do you need a side view of this one, or is just a
Yeah, I need a view of the underneath side.
Well,
underneath?
we'll look at a couple other ones and see.
He's not gonna move for us.
Nope.
Been looking at him for 15 minutes. There's another one,
Okay.
that one, right at the base of K.
Oops, went too far. Trying not to have the weight in the image here.
How can they do that?
Good shot, though.
It's like they know what I want to see and they're hiding it.
So the octopus in the lower left corner we suspect is a male, but we need to see one of its arms to confirm that. Octopus is hiding that arm from us.
Get that brighter just a smidge.
I'll hold it here to you.
That's good. Yeah.
I was just trying not to have anything washed out.
No, for sure, for sure. And that's been a problem with these.
This gives you a really nice view of all the water coming out of the seafloor here. This is a site of low temperature hydrothermal venting. Water coming out of these places has been measured up to 12 degrees Celsius. Okay, well, let's go see the other one.
Yeah, one of the reasons for males to be larger is that males fight with each other over access to females. These guys don't seem like they're fighting. Dead center there, please.
Do you think that scarring could be from another octopus?
I don't know.
No.
Once again,
Have you ever observed it? Them going at each other?
no.
Battle?
They kind of just seem really happy to be sitting in warm water.
Everyone's just relaxed at the
It's
spa.
like, yeah, hot spring. Where you been all my life?
One of the things you can see in this frame in the bottom right is what looks to be some green material on one of the rocks. So that's an indication where eggs had previously been attached, maybe at a different time.
Do they clean off a new area to attach to, or will they reuse some of that already?
They'll reuse it.
They'll reuse it.
All right.
Reuse it based on seeing the cement or like
So one of
The
the things we
same
want to do,
exact spot.
J-Rod, is get some of these shrimp with a slurp sampler. So there's a couple over here. I don't know if there's more.
There's some right down here.
Oh yeah, okay.
Yeah.
Right by the.
I'm gonna do just a little bit more video and then we'll try that before we pick up.
Now, I've confirmed on these like 25 females that 4 have eggs for sure that I've seen. So we're kind of assuming that the rest being in the same position are also brooding eggs. And they seem to have real tenacity to stay where they are. And that's another sign. If an ROV was poking me and I didn't have anything to defend, I'd get out of there. But if I had eggs, of course, I wouldn't. So that's our rationale. And other than poking every female with the temperature probe, I don't know how else to assess it. Just one of our stated assumptions.
Yeah, it is an assumption.
Extra footage.
With these rocks down here, Beth, with the cracks,
Yep.
they're not basalt, are they?
It's
you
hard to
tell?
tell.
Yeah.
It's hard to tell from looking. Like this thing on the left definitely looks like a basalt. I don't know
Right.
about those. Oh, okay. Okay. Let's just have a look at an octopus.
That male over there is starting to get a little
Squirrely.
active.
Oh, yeah, cleaning.
That behavior seems to be associated with some stimulus in the environment. The suckers have chemoreceptors, and like us, their epidermis grows and then sheds. So it's kind of like blowing their nose. They're clearing off the chemoreceptors.
Well, I
Or
hope
they're
someone.
just so happy to see it. I think they're clearing their chemoreceptors.
This is what an octopus blowing its nose looks like. I'll remember that. And is it going to show us its arm?
No.
Big stretch, big yawn.
It would be the third right one, so the one that's curled up behind
The weight?
the ones that stretched out.
It'll be this one back here.
Yep.
Oh,
Yes, I'll show you on the left side.
Don't want that in the frame. There's just so much stuff down here.
Yeah. Just thinking about it.
Yeah, it's just teasing.
Is there there's a different name for like the males and the female, like you got like a hen and a rooster?
No, we just call them male and female. We could come up with some terms if you like.
Okay, so let's go ahead and get set up for trying to get a slurp of some of these shrimp. If we still see some.
Save octopus lives. Slurp shrimp.
Yeah, so there's several down here. 1, 2, 3, 4. The BioBox 1 is still open. I don't know if we want to close that at any point.
This is my first start.
So see how this handle? Yeah, so this handle right here, it's pointing towards the arm. You can grab it at that angle.
Okay.
It really makes it easy for you to go out and manipulate. If you grab it straight down, you're going to be working with an arm like this. So,
In addition to the shrimp, we also want to try to get this crustacean.
squat lobster.
Yeah. We don't want them necessarily in the same canister. They might predate on each other.
Point out that little arm, like diagonally up in the neck. These octopuses are actually inside the seamount.
Oh yeah, they're inside every little nook and cranny.
Where they're safe from everything except the
But you can
shrimp.
get it with actual jumps at the core. So going a little bit deeper than that. I'll put it on a little bit. Now go down. Go down towards, down to the left. Just following the line. Yeah.
For anyone just tuning in, we are diving again at Dorado Outcrop, which is the site of our first couple dives on this expedition. You're currently watching live footage from remotely operated vehicle SuBastian, just shy of 3,000 meters below the surface of the Pacific Ocean off Costa Rica. And at Dorado Outcrop, we are returning to the site of several experiments that we left behind. We also collected a rock sample here, I believe, and we are leaving more experiments behind and samplers behind. And now the pilots are working to coordinate a little bit of collection here, hoping to collect some of these shrimp as well as possibly a little squat lobster.
Yeah.
Hola a todos quienes recién están sintonizando. Nos encontramos en este momento buceando en el sitio que se conoce como afloramiento rocoso dorado. Estamos a unos 3.000 metros de profundidad y este es un sitio que empezamos a bucear al inicio de la expedición porque sabíamos que era un sitio de anidamiento o reproducción de pulpos. El primer día o el primer buceo de la expedición dejamos algunos experimentos, así que hoy regresamos para revisar cómo estaban estos experimentos, también para colocar otros sensores adicionales. Hace un rato pudimos colocar un sensor que mide temperatura, otro que se va a encontrar recolectando de forma continua pequeños volúmenes de agua que van a ser procesados posteriormente en el laboratorio. También hemos tratado de observar en más detalle el comportamiento de los pulpos en esta zona y en este momento los pilotos del submarino están tratando de recolectar uno de estos camarones que hemos podido observar a lo largo de estos buceos que viven en una asociación bastante cercana con los sitios donde hay emanaciones de agua caliente y que es donde se encuentran los pulpos. Les recordamos que cualquier pregunta o comentario lo pueden hacer también en español. Nosotros tenemos aquí a bordo varios científicos que son hispanohablantes, así que igual pueden dejar sus preguntas en idioma español.
one little baby one right in front of you. What chamber, suction chamber are we on?
We are on 5.
Okay.
All right, let's have it go down. See if that's like it. These guys. Section is off.
Coming down to get this water.
When they start moving fast, just slow down. You gotta stay at 5 feet. Start moving back.
I'm coming for you.
Yeah, the ones that are coming from you, go for them. That's that. The one that's up. Right.
Why do they last this?
You go to your right. Going. Yeah, circle around, going to the forward. Going. There you go. There. Alright, I'll keep an eye up here, start working your way over to those guys over there. Once he comes in, I'm gonna turn the flow down quite a bit, so. He's in,
Okay.
goes in the down.
Para responder una de las preguntas que tenemos aquí en el chat, de hecho, los experimentos que nosotros colocamos en estos buceos, por ejemplo, las vasijas o las estructuras que ven de tubos de PVC con diferentes arreglos, fueron específicamente preparados para esta expedición. La mayoría fueron preparados acá a bordo del barco, entonces tomaron un par de horas para alistarse. Sin embargo, hay otros instrumentos que se han estado utilizando en todos los buceos, como los squeezer, por ejemplo, que son instrumentos que se utilizan también en otras expediciones. Entonces, lo que se hace es básicamente reutilizarlos y darles un mantenimiento para que puedan continuar funcionando en diferentes expediciones. Como les mencionaba hace un rato, en este momento lo que estamos es tratando de recolectar uno de los camarones que hemos visto que se encuentran viviendo asociados a estos sitios de emanaciones de agua caliente, donde también están los pulpos anidando.
Do you want to sit there in the boats in the middle so that I can direct them? Because before I needed to use the camera, but now I don't.
Sure,
Yeah.
because I can also share my profundidades. Okay, okay.
Once you get out of view,
That's it right there. You see its tail on this side there. Can't see it.
you can't see it.
Le agradecemos también a los hispanohablantes que tenemos en la audiencia. Para nosotros es muy importante saber que están siguiendo estas transmisiones y que nos están acompañando para aprender un poco más de estos ecosistemas de profundidad tan frágiles. Mi nombre es Celeste Sánchez Noguera, soy bióloga marina, investigadora de la Universidad de Costa Rica y los voy a estar acompañando por los próximos minutos.
You have them lined up, you know, I'm almost section.
So right now we are just like trying to sample one of these little shrimps. Most of you who have been following the previous dives are aware of the fact that these shrimps are actually living in a close association with these thermal fluxes. So we want to collect one of these organisms to actually being able to identify what is the species of this animal. Usually when there is like biological associations, they tend to be very species specific, which means that, for example, this particular species of shrimp is probably very related or specifically related to the species of octopus that we have brooding here at Dorado. El objetivo de poder recolectar estos camarones es poder identificarlos a nivel taxonómico correctamente, porque en realidad muchas veces cuando hay este tipo de asociaciones biológicas son muy específicas a nivel de especie, lo cual significa que muy probablemente esta especie de camarón en particular va a estar asociada únicamente con esta especie de pulpo que tenemos en este lugar. Entonces, también por eso es tan importante para nosotros poder recolectar el camarón. Perfecto, ya acabamos de agarrar uno. Ya tenemos algunos ejemplares de pulpos que recolectamos en el primer buceo de esta expedición en este sitio, y ahora al recolectar el camarón, entonces podemos realizar la identificación taxonómica y determinar si es una especie inclusive tal vez nueva que viva asociada específicamente en estas comunidades de pulpos.
With regards to dive time, we're looking at another 6 hours or so, somewhere in there.
Otro de los objetivos del buceo del día de hoy, además de colocar instrumentos de medición adicionales, otros experimentos que también queremos dejar, es tratar de comenzar a analizar más de cerca el comportamiento de los pulpos. Por eso fue que hace unos minutos buscamos algunos individuos que le parecieron particularmente interesantes a la doctora Janet Boyd, la especialista que tenemos a bordo en pulpos, y la idea de eso es tratar de documentar por medio de videos de una forma más cercana cuál es el comportamiento de estos animales. Recordemos que cuando estamos estudiando comunidades de organismos o incluso una especie en particular, para nosotros es muy importante hacer un análisis o un estudio bastante integral, no solamente a nivel de morfología, sino también a nivel de comportamiento, del entorno en el cual se encuentran viviendo estos animales. Y por eso es que también estamos recolectando muestras de agua, de sedimentos, para poder caracterizar cómo son las condiciones físicas, las condiciones químicas, microbiológicas de los ecosistemas en los que ellos se encuentran.
So you don't want to
Oh, that's
look
right.
for
We wanted
it.
to try that. If that spot lobster was still visible.
There's one down just below, all the way down in front of us.
El submarino SuBastian está equipado con diferentes instrumentos o herramientas que nos ayudan a poder realizar estas colectas de organismos. A veces utilizamos el brazo directamente, que funciona como un tipo de pinza gigante. Otras veces el brazo se utiliza más bien para manipular diferentes herramientas o instrumentos, como por ejemplo las redes que hemos utilizado en buceos previos para capturar algunos pulpos. Y en este caso el brazo lo que está haciendo es manipulando este tubo, que es básicamente como una aspiradora gigante que tiene el submarino, y entonces este tubo, una vez que aspira a los organismos, se conecta directamente a unos recipientes contenedores que están numerados. Entonces nosotros podemos saber si este camarón que se aspiró fue directamente al contenedor número 1 o número 2. Y eso nos permite también llevar un control bastante riguroso de cuáles organismos se están colectando durante qué momento del buceo y en qué lugar. Parece ser que vamos a tratar de recolectar esta langosta blanca que está en este momento moviéndose al lado izquierdo de la canasta. So we are going now for the white lobster that is slowly moving away from the basket. Let's see if we can catch it or not. Usually the diversity of animals living in these particular places where it's venting is very particular. So this is the reason why it's so important for us also to collect or sample these animals, because it's very likely that, for example, this species of shrimps or lobster that we are just seeing right now are not necessarily present in other places. It's very likely that they are actually restricted to these kind of environments. Es muy probable, como les comentaba hace poco, que estas especies, por ejemplo, de camarones o de langostas que estamos viendo en estos momentos, estén restringidas a este tipo de ambientes. Entonces, por eso es tan importante realizar la recolecta de estos organismos para realmente poder determinar si son especies que se encuentran también distribuidas en otras partes del fondo oceánico o si están restringidas únicamente a sistemas con emanaciones de agua termales. Si ese fuera el caso, significa que entonces es un motivo nuevamente de importancia para proteger este tipo de sistemas, porque albergarían una diversidad que es bastante única o bastante particular. El proceso de recolecta es bastante delicado porque pues en realidad tenemos que asegurarnos que no dañamos al animal cuando lo estamos recolectando, y por eso es que a veces si el animal se coloca en una posición en la que va a ser una recolecta, digamos, bastante agresiva, entonces se decide abortar y continuar buscando a ver si encontramos un organismo, un anhíbido que sea más fácil de recolectar.
I think it's to the port side, but 100% confident on that. But yeah, I think it's down.
You mind if we reference it when you
So it seems that we are going to try to find another white lobster that is in a more convenient position to be sampled.
Before we do this, when we get over to the next marker, there's hopefully an Osmo sample we're going to pick up and put in a spot. So,
it's just that thing's gonna be
yeah.
all the
Para aquellos que no nos han acompañado en los buceos previos, igual les recordamos que todas estas recolectas se realizan bajo todos los permisos otorgados por las autoridades nacionales de Costa Rica. Entonces, son recolectas también bastante planificadas, no recolectamos más organismos de los que realmente se necesitan, y todos los organismos que son colectados serán posteriormente depositados en el Museo de Zoología de la Universidad de Costa Rica, el cual es el repositorio oficial nacional, y ahí van a estar disponibles para toda la comunidad científica en el momento en el que se requiera hacer alguna consulta adicional en el futuro. Los pilotos en este momento están tratando de sacar de una de las canastas otro de los instrumentos que ocupamos colocar acá en el fondo marino. Pero el proceso de maniobrar a veces se complica un poquito. Realmente es admirable ver el trabajo que hacen ellos, la precisión con la que pueden a veces hacer ciertas cosas o manipular objetos debajo del agua. Algunos organismos pueden sufrir daños por cambios de presión cuando pasan de profundidades como la que tenemos ahorita a la superficie. Sin embargo, la mayoría de organismos que sufren estas afectaciones por presión generalmente son los que tienen estructuras que pueden, digamos, albergar o atrapar algún tipo de gas. En ese caso, las plantas o las algas sería el término correcto para los organismos productores primarios que podemos tener de alguna forma acá en el fondo. En realidad no sufren por el cambio de presión y para el cambio de temperatura. Sí, por ejemplo, cuando nosotros tenemos los organismos y en el caso de que algunos lleguen vivos a la superficie y ocupamos mantenerlos para evitar el estrés, precisamente los mantenemos en agua en un cuarto frío donde la temperatura es bastante similar a la temperatura del fondo, unos 2-4 grados. Entonces, con eso es con lo que se evita también como el shock térmico cuando llegan a la superficie.
Yes, we're going to recover the milk crate, and then where the sign of printing is, we'll do some temperature measurements. We will do a squeezer sample, and then we will try to deploy the temperature probe and also the microcolonization experiments that are all on that front firebox.
So we are doing all that in the marker R. Okay. Okay. So, como pudieron observar, hay diferentes letras. Estos son los marcadores que hemos colocado. Algunos se colocaron en la expedición del 2013, hace 10 años, y otros se han colocado ahora en el primer buceo que hicimos hace un par de días cuando comenzamos la expedición. Y básicamente nos sirven como eso, como marcadores para que cuando regresemos al sitio podamos saber con exactitud, ok, estuvimos haciendo tomas, observaciones o colocamos instrumentos cerca del marcador R o cerca del marcador W. Es la forma que tenemos nosotros de poder ubicarnos nuevamente en el sitio que estamos estudiando. Entonces ahora lo que vamos a hacer es trabajar un poco alrededor de este marcador R, donde se van a hacer nuevamente mediciones de temperatura. Se va a dejar además un sensor que va a estar midiendo temperatura de forma constante de aquí hasta la expedición de diciembre, y también vamos a colocar algunos experimentos de asentamiento como estos tubos de PVC que ven a la derecha de la imagen. Y si no me equivoco, creo que tal vez también vayamos a dejar otros experimentos de asentamiento con otras formas y con otros materiales.
anything in particular you want to hide or zoom on, or?
I think I forget.
So good.
So we can go ahead and take out.
Uno de los objetivos de colocar este tipo de diferentes estructuras es precisamente para identificar si los pulpos tienen una preferencia de uso del sustrato. Parte de lo que se cree, o una de las hipótesis que se manejan, es que tal vez están utilizando el Dorado como un sitio de anidación, no solamente por el hecho de que hay emanaciones de agua caliente y esto podría ayudar a acelerar el proceso de eclosión de los pulpos, sino también porque en realidad hay mucha disponibilidad de sustrato, que en este caso sería rocas y rocas bastante limpias de sedimento. Entonces, al colocar nosotros, por ejemplo, diferentes materiales con diferentes formas, tubos de PVC, unos más pequeños, otros más grandes, también estamos colocando vasijas de barro. Todo eso nos permite, cuando luego regresemos, ver si realmente los pulpos lo comienzan a usar. Y si la preferencia es porque sean lugares con emanaciones de agua más caliente, o únicamente por tener a disponibilidad un sustrato libre de sedimentos para poder colocar los huevos. Por ese motivo también hemos colocado o vamos a colocar algunos de estos experimentos lejos de donde están las emanaciones de agua tibia, pero relativamente cerca para que los pulpos todavía puedan llegar a este lugar. So right now we are just going to proceed to deploy some of these experiments or colonization experiments here close the marker R. Remember that we were working previously was in marker W and K, if I'm not wrong. And we are just going to repeat exactly the same procedure. So basically measure the temperature, deploy the probe temperature, deploy some of these experiments as well. And we are using these markers with different letters just as a way for us to actually have a good orientation here in this study site.
There's a Janet. There's a really nice shot from an earlier dive. That's like the whole view of marker R, W, and K. There's at least 200 in the shot.
You're a kid.
I was like, I can't keep track of that.
It looks to me like there's 3 on the middle crate.
Yes.
That could be the middle track. If it's here, it's got polka dots on the screen. So you may not.
Les recordamos que el día de hoy estamos buceando en el afloramiento rocoso Dorado, el sitio que se exploró por primera vez hace 10 años y donde se encontraron los pulpos asociados a las emanaciones de agua caliente. Y en esta expedición, el primer día de buceo lo hicimos acá en este sitio y hoy estamos regresando nuevamente para poder revisar algunos de los experimentos que dejamos hace unos días y poder colocar experimentos nuevos y colocar sensores nuevos. Así que el buceo va a continuar por un par de horas más, aproximadamente hasta las 7 de la noche, hora local. Así que si saben de alguien que le pueda interesar tener esta oportunidad única de ver pulpos a 3.000 metros de profundidad en un monte submarino costarricense. Los invitamos a que compartan el link de la transmisión con estas personas. Para aquellas personas que están comenzando a ver la transmisión en este momento, les volvemos a comentar que en este momento nos encontramos colocando algunos sensores y algunos experimentos en este sitio de anidación de pulpos que es el Dorado. También es interesante observar cómo se pueden distinguir, en este caso, las hembras de los machos. Las hembras son todas estas que se encuentran en una posición como boca abajo, porque realmente ellas adquieren esta posición porque lo que están haciendo es protegiendo los huevos. Entonces se colocan de esta manera y también por eso se quedan bastante estáticas, no tienen mucho movimiento. Recordemos que los pulpos son un caso bastante interesante de reproducción donde hay una única, digamos, reproducción a lo largo de su vida. Una vez que ha ocurrido la cópula, las hembras proceden a colocar los huevos en un sustrato, que en este caso son las rocas, y en ese momento dedican todo el resto de su vida a proteger los huevos hasta que eclosionan los pulpos pequeños, los pulpos bebés. Entonces, una vez que los pulpos han eclosionado, la hembra muere. Generalmente durante todo este tiempo que ella permanece cuidando los huevos, deja de comer, entonces se debilitan muchísimo. Lo que hemos podido observar en este sitio es que los machos parece que tienen un tamaño más grande que el de las hembras. De hecho, lo pudimos comprobar con los especímenes que recolectamos en el primer buceo. Este que ven aquí, de hecho, en la canasta que estamos moviendo, al lado derecho es un macho. Entonces, una de las formas de reconocerlos es porque son evidentemente más grandes que las hembras y porque generalmente se encuentran en una posición más abierta, con el manto extendido. Incluso algunos se les puede observar moviéndose entre las rocas, a diferencia de las hembras que, como les comentaba, se mantienen en una posición bastante estática. El tiempo que duran en eclosionar los huevos varía dependiendo de la especie. En estas especies de profundidad se cree que pueden durar varios años y es algo que todavía no sabemos con seguridad cuánto es el tiempo para esta especie en este sitio, pero sí pueden ser varios años. Entonces, realmente es bastante interesante saber que se pueden mantener tanto tiempo igual vivas sin tener ningún consumo de alimento. Y precisamente como es un periodo de incubación tan largo, cualquier aceleramiento de ese proceso que puedan tener por el leve aumento en la temperatura del agua va a ser una ganancia.
So the plan with what's underneath and
You said? Yeah. Can we put the fauna associated octopus stream? Sure. Yeah. Plan as it goes on the basket. We'll try to like grab and just like put it aside. There's a plan. There's obviously an octopus in the way in both places. So probably we're trying to put it on the note plate if we want to just gently balance it over the Excalibur to see if that encourages the octopus on the bottom of the month. And then, we reach for that black cable and see. It's not well.
Esto que estamos viendo en este momento es lo que estamos haciendo es básicamente recuperando uno de los instrumentos que habíamos dejado en este sitio. Entonces, lo que están haciendo en este momento, una de las colíderes de la investigación de la expedición junto con los pilotos del submarino es tratando de ver cuál es la mejor manera de manipular esto y de colocarlo de regreso en el submarino. Recuerden que en el submarino tenemos una plataforma con diferentes compartimentos y canastas destinados para llevar todos los instrumentos que tenemos, pero también todos los organismos que recolectamos. Entonces realmente es bastante importante a nivel logístico organizar cómo es que van a ir todos estos instrumentos cuando los ponemos de vuelta en el submarino también. Y nuevamente le agradecemos a todas las personas que están conectadas en la transmisión. Para nosotros es muy importante poder aprovechar este tipo de oportunidades para que las personas puedan aprender un poco más sobre nuestros fondos marinos. So right now we are just recovering the Osmo sampler, trying to figure out what will be the best way to actually put this back on the SuBastian. Parte de lo que justo estaban tratando de ver o estaban discutiendo era cómo deshacerse de algunos de los pulpos que están en el instrumento que vamos a recuperar, cómo espantarlos, pero en algunos casos hay otros que pues si ya están ahí metidos probablemente se van a venir en la canasta y van a hacer una colecta digamos secundaria a partir de la recuperación del instrumento de medición. Acá pueden ver en la imagen un poco de lo que les comentaba de los diferentes compartimentos que tenemos en la plataforma en el frente de SuBastian, así como algunas de las herramientas y los instrumentos que utiliza el submarino para las diferentes labores científicas. Les agradecemos yo me despido de momento y le voy a dar el campo a mi colega Gustavo, quien va a estar con ustedes las próximas horas, al menos de la transmisión. Gustavo también es bilingüe, así que pueden continuar también con él evacuando algunas de las dudas que tengan al respecto. So I'm now going to step aside and going to give the spot behind the mic to my colleague Gustavo, who will be with you for the next hour of this dive. Thank you very much. Muchísimas gracias y pura vida. Yeah, because we are just like actually starting our marker out, so it's like Osmo rotary, and then we have to continue credit at whatever it's happening. And so yeah, yeah.
No, he's still viable here.
Yeah, of course. Yeah.
You want us to fly?
No, if he's flown away, we'll try to get another one.
He's in the front porch.
He's in the front porch? Okay.
Like a mad possible. Now
Oh, I see. Yeah, if you want to fly away to do that, that's fine. Let me know if I can help with the camera. Is it okay if I move the camera over?
I'm going to start thrusting down here. Can you get net behind it? Push forward. Ready? Push it forward. Watch the bow lines. Just come out of there for a second. Let me see if I can get him to come off.
So one of the reasons we're interested in collecting this particular specimen of octopus is that we believe it's a male. We've seen indications of that with its arms. We've only collected, I believe, 1 or 2 male octopus on this expedition so far, but it's important to have examples of both species, both sexes, for understanding the species, its biology, its reproduction. All of the samples that we collect will be stored at the Museum of Zoology in Costa Rica, where experts from around the world can come research them or share those materials. So right now we're flying the ROV up into the water column so that we have a little bit more distance from the seafloor, and trying to use the currents that generate it as we move around to encourage the octopus to come into our sampling.
Going after this one first.
No, well, excuse me.
I definitely don't mind that.
He seems to be well anchored to that spot.
Try it one more time. If you want.
Yeah, I think one more time, and then we'll go back to our operations and
This time
do
go
a
to
more
that side.
intentional collection.
Gonna come up, get up in the midwater, and then this time I'll push forward. Just stay like further away from the back. Just get in this little gap right here. And rotate. Yeah, just like that right there. Kind of. All right, ready? I'm gonna go forward. Yeah, he's not gonna do it.
No.
Back down. Get your arm out of that. Comes out later. What's that? Okay, go ahead and just come out of it so I can get that tray and so. Okay. See what I'm doing. See if he comes
So
out.
we can just mark that we have a potential octopus on the back of the basket. It's not necessarily sample, just in the books. Because it would have been attached to the Osmo sample recovery. So it's not on there yet. So that recovery should have a sample. Yeah. Anything that's recovered that's a need for recovery. Did it not ask for that in the deployment under recovery menu? Okay. Is it okay if I move the camera a little bit? Or are you using it to fly? Okay. Gus, are you on?
For sure.
Yeah, just for the camera.
Heading back to Mark.
Yep. So that's so it's over to our port side. There it is.
So we're gonna wanna try to find a metal. That one right there?
Yeah, so that's not the priority operation right now.
Okay.
I would like to go back to what we were doing.
Okay.
So we'll put the net back. It's more just opportunistic to try to get that one off. Great. So it's still lying back there.
Kind of roll it up.
For those of you just joining us, welcome. My name is Gustavo Ramirez. I'm an assistant professor in microbial ecology and computational biology at the California State University in Los Angeles. I am joining the control group in the control room by our expedition's co-chief scientist, Dr. Beth Orcutt from Bigelow Laboratory for Ocean Sciences in Maine. And I am also joined by a graduate student from the Calgary, University of Calgary in Canada, Mr. Rob Perrin. The three of us are part of represent the science party for an amazing expedition. We are on board the research vessel Falcor 2 that is owned and operated by the Schmidt Ocean Institute. And along with us here, about 150 kilometers away from the Costa Rican port of Punta Arenas, we also have with us an incredible piece of equipment called SuBastian. So that's what you're seeing on your screen here. SuBastian is a remotely operated underwater vehicle that comes along with its friends. So there's about a dozen engineers and pilots that are on the ship here with us too, that are able to deploy SuBastian from a cable on our boat here and navigate this remotely operated submarine down to a depth of approximately 3,000 meters, 3 kilometers. So that's about 12,000 feet beneath the waves, where we are studying a series of seafloor piles of rock or features that can be pretty big in some instances when they're called seamounts, or they can be slightly smaller. That are remnants of volcanism in the area. And what drives us here specifically is the fact that previous expeditions that were here about 10 years ago identified very interesting octopus behaviors in the area. So basically, the seamounts, they funnel a lot of this deep water amongst them, and this funneling is based on the physical property of density and like the porosity of the rocks. So slightly warmer rocks. At slightly warmer sites, the water that's flowing underneath the crust, which is under the sediments, is released. And so where those seamounts release water, that tended to attract a disproportionately high number of octopus females. The females then tended to proceed to lay egg clutches or bundles of their eggs right where this shimmering fluids emerge from the rocks, and this very, very interesting behavior was not observed before, and we are here to study how that works. That is exactly what you're seeing here on your screen. We are on the south western side of an outcrop or a seafloor feature called Dorado. This is the place where these gigantic, by the hundreds, if not thousands, octopus nurseries were observed during our first visit here over 10 years ago, or around 10 years ago. And there was a couple of salient questions that remain after El Dorado octopus nurseries were first visited. First of all, despite seeing these females, which are basically folded inside out in a perpetual embrace of their egg clutches, despite seeing these females taking care of their eggs right at the sites of venting fluids, it was unconfirmed as to whether or not their eggs were viable. So it was kind of a big big question that remained when we last visited this site 10 years ago. Are these females brooding viable eggs? There was a lot of egg close-up 10 years ago, and it was really hard to tell if those eggs were viable. So it's kind of one of the first order questions that we had during this expedition, is to verify the viability of the eggs in the octopus egg clutches here. So during our first dive to this site over a week ago now, we were able to confirm the viability of the eggs. We basically had really good close-ups of dozens and dozens of clutches that appear to have embryos looking right back at us. And this was confirmed just a few minutes later when looking at the brooding females. We just started to see at first a few, and then after a couple of minutes, a couple dozen octopus hatchlings swimming right in front of our camera. So it was a very exciting point during our first dive that basically answer a major question, major lingering question about the ecology of and more specifically, I guess, the behavior of octopuses in this extremely remote habitat.
Exactly what she said. Yeah. So the bridle is.
And we'll just want to, turn the manipulators of the octopus that are attached to the outside and facing outward.
The bridle has a black. Yeah. You see it.
Yeah.
So what you're seeing here is the collection of an osmotic sampler. So this is a very interesting piece of geochemical equipment that allows us to collect, continuously collect small quantities of fluids in a 1 millimeter diameter line that can be hundreds of meters in length, just coiled up. And this is basically a pump again that brings in a known amount of volume into a small coil. And this movement of water is powered by osmosis, as the name would imply. So there's a pump in this crate that contains a brine separated by a semi-permeable membrane from DI water. And the differences in water content between the brine and the DI water drives the uptake of what's initially water in a very long coil, or DI water in a very long coil, that's slowly displaced by environmental samples, so water all around it. We know at what rate that water comes into the coil, and therefore we know how much water is displaced as a function of the length of the coil. Therefore, after an experiment is sitting at the seafloor for years sometimes, we're able to section the coil accordingly to then keep track of the geochemical profiles with a timestamp and basically get a video of the changes in geochemistry, monitor continuously at any site in the ocean where one of these osmotic pumps are deployed.
You want me to get rid of it with the magnet?
Well, at some point, we're going to want to turn the milk crate counterclockwise about 90 degrees so that those other octopus are facing outward.
Yeah.
And so I'm not sure if when we bring it in, if that other.
Yeah, I mean, but this one, you want me to try to get it off?
Yeah. Let's see if she'll come off.
Oh, I like this.
Well, she attaches to us, we can put her back on a rock.
So again, we're trying to make this osmotic sampler collection here, but looks like we've got a couple of hitchhikers. We are extremely mindful that we need to minimize the impact that our presence and work at the seafloor has on this very unique communities. So we're trying our best to leave behind the hitchhikers here. and only collect the our intended collection, which is
No, you're fine. You're fine.
which is a scientific piece of equipment.
Is that a male?
I can't tell.
I don't think so. No, it doesn't have a different arm.
You're just zooming out, or?
Right. And do you want to do that with the other ones, or just?
No, for those, I think if we just kind of turn the basket counterclockwise now, about 180 degrees, and they'll be facing outward, and then they can decide to liberate themselves or not.
I'm just going to get this out of your way while you do that. Sounds good? Yeah.
So we are near a place called Marker R and actually Marker W2, pretty close to each other.
Yeah, the basket itself.
These sites were identified as sites of relatively high rates of hydrothermal discharge on this seafloor feature called Dorado Outcrop. When the site was first explored, close to 10 years ago now.
You want to zoom in on that, or you got it? Okay.
Doesn't seem like it wants to move. Yeah, but I can't get over there. Yeah. We'll see if we can get it to fit in, and then we'll try to move it. Okay. If I have to, I'll grab that bridle from you to try to push it in. I just don't want to push too hard that way. Yeah.
Just to give everyone at home a sense of the action that's happening here in the ROV control room. Again, what you see on your screen is the work basket of a remotely operated vehicle, and whatever happens down there needs to be whatever happens down there is piloted by two very skilled pilot engineers that are sitting here in the control room. So they after hundreds of hours of practice, thousands of hours of practice, perhaps, they're able to be they're able to have extreme dexterity with what the robotic arms can do at the seafloor, and all of that again is mediated by basically playing with this really weird looking joystick happening right next to me. So again, we're attempting the recovery of a scientific instrument called an Osmo pump, deployed here many years ago. And basically what you hear in the background is the pilots communicating regarding movements and the rearrangements that need to be made on the recovery payload bay of the ROV to fit this large milk crate that hosts this geochemical instrument.
I can lift up on this bridle shoulder bring yours in as well. Now when you get closer to quiver 6. And then once you get that, start coming down on your side. Start coming down. I'm gonna hold there so you can get it to this level. You try to push down on that back corner a little bit.
Corner closest to us or this?
Further away.
Yeah. I don't even need to let go of this middle bit.
Okay.
I think it's in business.
Go ahead. I think I got it. Try moving the tweezers. Yeah, take that starboard forward corner, pull that back. Go outward a little more. You gotta go out a bit.
Out?
I think so. I think if you go out, no, sorry, no. Outward.
That worked.
Yeah. Okay. Wow.
Teamwork makes the dream work.
Good job, guys. Wow.
Now we gotta, you may do the hoses since I got the intermission.
Yeah, you can do. You're at this. I don't know the other.
We're gonna bring the porch in a little bit.
And so we just witnessed the recovery of a milk crate housing a pretty interesting piece of scientific equipment here called an Osmo sampler. Again, we have to thank our ROV team for doing all the small scale maneuvering there that was required to fit this milk crate right in the middle of the payload bay. It's pretty snug there, but it worked nevertheless. There appears to be 2 additional hitchhikers on the outside of that milk crate that should be able to fly away as soon as they choose to do so. Well, we still await this longer sampling line. I guess I'll take some time to give everyone a little bit of a big picture perspective of why we think that this site is incredibly important for understanding not only local processes with the charismatic, beautiful octopuses that live and brood their eggs here, but also at super important for scientific knowledge at the planetary scale. So what we see here is sites of relatively low temperature hydrothermal fluid venting. And this venting is water that has percolated into the upper layer of the crust, moved for long distances in some instances under a layer of sediments, and again, powered by slight differences in temperature in the first layer of the lithosphere, covered by sediments. Those slight differences in temperature basically has evolved. So the bulk water that comes in at places called recharge sites that are slightly colder than places where water dissipates or exit sites, the cold water that comes in is biologically, geochemically, and physically identical to background seawater. It's literal background seawater that enters the crust at recharge sites. As that background seawater flows through the crust, it changes physically, geochemically, and microbiologically, and transforms or evolves into hydrothermal fluids. Those hydrothermal fluids carry with them a completely different geochemical signature. And what I'm most interested in as a microbial ecologist, interested in microbial activity in the crust, those hydrothermal fluids also carry with them distinctly different microbial communities relative to the microbial communities of the background ocean where they may end up discharging. So that's the piece of the puzzle that I'm really the most passionate about here. Interestingly, however, this low temperature fluid movement under the crust, the first layer of the lithosphere, rocky part of our planet, this phenomenon cycles the entire water column of the ocean with a periodicity of about once every 300,000 years. And this cycling has extreme repercussions for determining the equilibrium chemical state of the planet. I guess the ocean more directly. Then the ocean interacts with the atmosphere, and that interaction, driven again by this water flowing through the cracks in rocks underneath the sediments, can has the potential to ultimately determine really important things at a planetary scale, such as the oxidation reduction state of the Earth. We know from the fossil record that when that number has changed, that has triggered major changes in which lineages evolve and which lineages don't. So we know that there's this intimate interrelationship between just basic physics of the planet, emerging geological and geochemical properties that then dictate those properties, very important ones, again, like the oxidation reduction state of the planet, that then guide the entire parade of evolution on this planet for all animal life. So it's a super interesting connection that I always like to remind the audience about, and that's really the motivating factor as to why I find myself here. A couple of other activities that are planned for the next hour or so of this dive are the collection of pristine fluids from hydrothermal venting in the rocks. We plan on doing this by using something called blanking out on the name, just one second. Squeezers. Yeah, so we will collect the fluids using really cool pieces of equipment that we call squeezers. And what the squeezers are basically really sophisticated syringes that allow a plunger to be retrieved by having the ROV manipulate triggers with at the seafloor that basically move a plunger back and displace what's originally in there, which is the eye water, with environmental sample. Or again, if this is performed next to a charge of next to a site of hydrothermal discharge, then you end up displacing that DI water with hydrothermal water, pristine hydrothermal water, which is not easy to get. Very few places in the world where a sample like that can be collected, and this is one of them. One out of two, actually, as far as I'm concerned. At some point during this dive, I also expect to deploy a microbial colonization experiment at a site of hydrothermal venting. I've deployed 3 of these experiments during this expedition so far. The first one was done not that far away from here, perhaps a few meters away from where we're at right now.
I just went back to the first dive wrong and saw that it was us that put it back in.
So the second experiment I've deployed at a place without venting, and the third experiment I deployed at a seamount that we visited 3 days ago. That is the second that has the that also has hydrothermal venting and octopuses brooding around the rocks where the warm fluids emanate. So that's where I deployed a third experiment so far. I hope to deploy a fourth today, but this fourth experiment will be a second experiment on the Dorado seamount that we're visiting today. So each one of my experiments are comprised of 5 sterile mineral substrate types. Each one of these substrate types is deployed in its own 5-pound dive weight. That it's basically has a PVC housing that contains wrapped up in pretty fine mesh 30 to 60 grams of a single sterilized substrate. And the goal here is to see if the substrate type plays a role in the kinds of microbial communities that colonize those initially sterile surfaces over a deployment period of 6 months. So in December of this year, myself and many of the current Octo Odyssey expedition members will return to this site and retrieve experiments that are left behind this summer. So after 6 months of incubating this initially sterile rocks at sites of hydrothermal venting, I will be able to then extract DNA from each one of these substrates to have a look to whether or not there's differences that are driven by the different substrate type deployed at each one of the venting sites. I will also be able to have cross-venting site comparisons where I can compare one communities on the same substrate at one seamount where there's venting versus another seamount where there isn't venting, or a control, basically the same substrate deployed at a place with no venting at all. So these kind of first order ecological comparisons will allow us to assess the effect that the distinct microbial communities that live in the crustal fluids have potentially on the weathering of the rock, and also very importantly on the biology all around the venting areas.
Do me a favor and shoulder left, see if you can push it in. Oh, you're out, okay, you're out of there. Yeah, see if you can just push that loop underneath it a little bit better. That's usually right there. Okay. Yeah, if you go too much, it's gonna pop all the way out. Hold that. I think we'll give that a go. What do you think? Beth? Do you think we're good?
Yep.
Hopefully it doesn't fly around. All right. What would you like us to do now then?
We want to get the T-probe to do some temperature querying of the venting fluids.
In this area right here?
Yep. And then we'll try to get some squeezer samples after that.
Okay. We'll just go and grab the different now.
Yeah, we can.
What was that?
So with the osmotic sampler safely recovered now and safely stowed away in our ROV payload chamber, we are moving on to doing direct temperature measurements of sites of potential heat flow amongst the immediate vicinity here. So we'll be looking really closely at rock fractures and looking for shimmering. And whenever we do suspect shimmering, we will take additional temperature measurements.
We recovered one on the second time.
Buenas tardes al mundo de habla hispana. Me llamo Gustavo Antonio Ramírez Ábalos y soy un profesor asociado en la Universidad de California, Cal State Lake, en Los Ángeles. Me encuentro a bordo del buque de investigación Falcor 2, que es coordinado por el Schmidt Ocean Institute. Y bueno, el buque y un grupo de científicos internacional nos encontramos aproximadamente a 150 kilómetros adentro del mar del puerto de Punta Arenas, Costa Rica.
Yeah, so we want to go interrogate. Basically, we're on the Octopus Odyssey.
That's cool. So just get over by R.
Yeah, exactly. So we'll do some in there. We'll also do some by where the PVC tubes are, so we can do them in either order. Before we do that, we're just going to tight zoom on that little wiggly one.
Keep doing that behavior.
Yeah. It's
Tighter than that.
Shedding that skin.
Let's hold this for just a few seconds. Yep.
I mean, those shrimp are bold. Yeah. Because, octopus, a lot of them can be a predator, so.
Yeah, and then just a little bit tighter on that octopus. Okay, so
Nice. Wow, that's cool.
Get out of here.
you can see there's some eye spots in that, some of those eggs. Just protect them.
I can get it. Might be able to get a tighter zoom on that too.
No, it's okay. The action is the protection. So what that is. Okay, that's good.
And then go get into that area pretty much.
Yeah, so however you want to set it up, if you want to do the PVC traps first and then the stuff.
I'm gonna try to get us in a position to where we're able to get all that
Okay.
from the same area, the same zip code.
Buenas tardes de nuevo y sí, nos encontramos en el buque de investigación Falcor 2 a 150 kilómetros del puerto costarricense de Punta Arenas. Y lo que estamos haciendo aquí es que estamos formando parte de una expedición que se llama la Odisea del Pulpo, Octopus Odyssey, patrocinada por el Schmidt Ocean Institute. Y bueno, nos encontramos en en este buque con un grupo internacional de científicos, oceanógrafos, biólogos, físicos, geoquímicos, microbiólogos. Y, y nuestra misión es de entender la biología que ocurre en montañas en el lecho marino que se llaman seamounts, donde emanen aguas hidrotermales. Estas aguas hidrotermales parece que atraen a hembras de esta especie de pulpo profundo, y estas hembras justo solamente, casi exclusivamente donde hay aguas hidrotermales que salen de estas piedras, dejan sus huevecillos y se dedican a incubarlos. Y es lo que estamos viendo aquí en la pantalla. Pueden ver docenas, pero bueno, si viéramos todo el panorama, hay cientos o si no miles de hembras que tienen de 6 a 12 huevecillos debajo de sus cuerpos, y básicamente los están abrazando y protegiendo de los depredadores naturales que se encuentran en el área. Esos depredadores son los camarones que podemos ver ahí justo al lado del marker R. Esta actividad en los pulpos no se conocía hasta que se exploró este monte en el lecho marino. Y bueno, este descubrimiento se hizo aproximadamente hace 10 años y es la primera vez que regresamos después de 10 años a explorar lo que está pasando aquí. Durante el reporte preliminar hace 10 años, se vieron las hembras, se vieron los huevecillos, pero no se había confirmado si los huevos de pulpo eran viables o si había verdaderamente embriones. Nunca se pudo ver un embrión cuando visitamos este lugar hace 10 años. Contrario de eso, nuestro primer buceo durante esta expedición hace más de una semana ya fue en esta área, y lo primero que hicimos es tratar de investigar si los huevos de los pulpos estaban viables o eran vivos, y después de acercarnos, hacer varios acercamientos con la cámara de alta resolución, pudimos confirmar que en realidad los huevecillos tenían embriones que nos estaban viendo también a nosotros. Unos minutos después pudimos ver que había pequeños bebés de pulpo que acababan de nacer en todos lados, por docenas. O sea que eso ya es un descubrimiento muy importante que hemos podido hacer durante el primer buceo de nuestra expedición. Bueno, no estoy seguro si haya hispanoparlantes en el chat. Cualquier otra pregunta en español, e incluso en inglés o en portugués o en alemán, estamos aquí para responder sus preguntas. Voy a regresar a dar el comentario de lo que estamos viendo aquí en inglés, pero bueno, obviamente si hay alguna pregunta, cualquier cosa en español, yo estoy aquí para responderla también. Cabe mencionar que la mitad de los científicos en el buque son hispanoparlantes. Obviamente estamos en aguas territoriales de Costa Rica, y es extremadamente importante para los costarricenses que podamos explorar estas aguas profundas de su territorio, que verdaderamente el 92% o más del territorio de Costa Rica es justo donde estamos ahora. Son partes del lecho marino profundo. O sea que es súper importantísimo para nuestros colegas ticos entender la biología y los procesos ecológicos que ocurren en estas profundidades para poder verdaderamente entender la mayoría de la naturaleza en su territorio nacional.
That's where it's coming from. Yeah. So.
Wow.
Let's try to get. Yeah, let's hold this shot for a moment. And kind of in that gap between those two at the bottom. We'll try there first, and we'll try to come above the octopus.
Thank you.
develop this one gets a little bit better. And like develop it so that it's more pointing in the same direction.
Quite an incredible shot that we've got here. Plenty of hydrothermal fluid discharge. I like to remind the audience, though, that even though this looks very hot, it's only slightly above background temperatures. Well, I guess maybe perhaps 10 degrees above background temperatures. We'll find out when we stick a thermometer right into this crevice and get an actual measurement. What we see in the lower left-hand side there is obviously a female that is brooding her eggs. And to do this, they just undergo this, what it seems to be a perpetual embrace. I'm not sure what happens to the mothers after sitting here for years, taking care of their eggs. The hypothesis is that after taking care of the eggs for years, they might simply senesce. Yeah, again, this is quite an incredible site. Plenty of fluid recharge and just octopus everywhere. Some of the most interesting hypotheses that we're testing in this expedition regarding octopus behavior aim to truly understand the drive behind their brooding site preferences. What exactly drives these females to lay their clutches here? Is it the fact that there's just simply barren rock and the hydrothermal discharge by exerting force basically cleans up the surfaces of this rock so that they never get sedimented from the perpetual onslaught of marine snow from the water column above? So that's one hypothesis, that females are not particularly happy with the temperature or the component of the hydrothermal fluid, but are basically taking advantage of sites where the fluid never goes away to ensure that their eggs don't get sedimented on. That's one potential hypothesis. Another hypothesis is that there's something in the fluid that perhaps helps with metabolism or with the development of embryos in the X-axis. Third hypothesis, and as a microbiologist, for me, like the third hypothesis is the most sexy. It could be that there are microbes that come out in the fluids that play a role in the actual development of the embryos and or a role in the physiology of the females. Yeah, so reiterating this third hypothesis is that the microbiome, the unique microbiome that's present in the venting fluids, may be playing a role in the brooding preferences of this the female octopus here. Again, perhaps serving or providing a service as a probiotic to either the brooding mothers or the developing eggs and embryos. And of course, a fourth hypothesis could be that all these three hypotheses combined play a role play the most important role in the behavior also. So we really don't know, but that is what we're here to find out.
Reading in between these two. Okay. And then potentially. Do I say anything? Okay. I'm going to sign up for so are you next? We'll say for in front. Yeah. So it's right there. Yeah, I think that's what we do. Oh, yes.
Where did my camera go?
I will be signing off now, and in my place, you guys will hear from Professor Maria Sandoval of the University of Costa Rica.
Sorry about that. Had our
Again, my name
cameras,
is Gustavo
go by.
Ramirez, assistant professor at Cal State University in Los Angeles, and it's always a pleasure to share the excitement of and wonderment of this exploration that we get the privilege to enjoy daily on board the Falcor 2 research vessel as part of this Octopus Odyssey Expedition. So again, I'm signing off now, but you will be joined live with expert commentary by Professor Maria Sandoval from the University of Costa Rica, my colleague here.
Sorry about that momentary blackout on our camera.
We just collected Osmo sample. I think that was one of the old ones. It was here from the last expedition, and so that took a while. So the octopus wouldn't let go. Now we're in the middle of getting more temperature measurements. We're going to go in the crevices. At some point, I'm not going anywhere until we deploy my experiments, so I'll be in the paper.
We got octopus on the board.
I did more Spanish and
Yeah, I came up with our milk crate, and there might also be a here
Good afternoon, everyone. I'm Maria Sandoval from University of Costa Rica, and I will be here with you asking your questions and telling you about this expedition and this visit again to Dorado Outcrop. And so if you have any questions, you can ask me also in English or in Spanish. Hola a todos, a todas, es María Isabel Sandoval de la Universidad de Costa Rica. Estaremos aquí, estaré esta hora con ustedes hablando y contándoles un poco de nuestro regreso al sitio El Dorado, este sitio que ha sido en particular una de las principales objetivos de investigación y que nos ha traído, ¿verdad? Ha traído esta expedición internacional hasta nuestras aguas acá en Costa Rica. Sí les comento que me pueden hacer sus preguntas en español o en inglés, si desean. Y estaremos compartiendo un poquito de lo que hemos hecho y de lo que seguiremos haciendo. Aquí lo que estamos viendo, así como se ve este agua, es un agua distorsionada que le llamamos, que es la que indica que hay un flujo de calor, digamos, varía con respecto al agua circundante. Aquí está saliendo un poco de agua más caliente. Recordemos que los valores en este momento están tomando unas medidas de temperatura y llegamos a unos 9 grados acá. Esto ha sido una de las hipótesis también para decir que acá es estas temperaturas, digamos, colaboran o pues es de las principales razones tal vez por la que lo que son los pulpos vienen a anidar acá.
Mike, if you want to kind of bring it up and behind that octopus a little bit more.
So here what we are doing is to try to get some heat, some temperature measurements, and so we are trying to do the best, as close as the octopus. We are taking these heat measurements or temperature measurements from the deepest part of the shimmering water. So they're trying to do that.
I'm watching your, I'm watching the porch.
All right, 10.08, 11. You're getting right in there.
Que tenemos por acá principalmente es, bueno, aquí tenemos 12. Wow.
Fortunate part is I can't get a temperature probe to go up there. Temperature logger. But that's the highest temperature we got last time. So that makes sense.
Aquí estamos viendo una de las máximas temperaturas que
Go ahead and pull back a little bit
hemos
and we'll find another spot.
medido en lo que es el sitio del Dorado. En esta zona tenemos variaciones de temperatura. Hemos medido entre 9, 10 y aquí es donde estamos teniendo 12.2, ha sido la más alta.
This lighter colored area down here, kind of in the center of the frame, if there's any warmth at all there, just because I'm trying to think where we're going to put Gus's experiment. I don't think we can chuck everything back in that hole.
See the red dead center? There's like below that center octopus. There's kind of like a light.
Podemos ver bastante vida circundante relacionada a lo que son los pulpos, por ejemplo, camarones, esponjas, poliquetos, estos organismos como gusanos que se ven ahí, carbonatados, estos son lo que llamamos poliquetos. So what is generating the heat normally is that we have water that is coming in the crust and passing by through the crust, and then it's like hitting in this way through the crust, and then is coming out like with a little bit of we can say warmer water that was in the side that was getting into the crust, like normally is colder. So that's why we are in those sites that we have is some seamounts and some outcrops that are the biggest ones that usually are getting the cold water into the crust and going out in the smaller outcrops like this one, like Dorado, that is getting the outflow a little bit warmer. So that is the idea of what is happening here. Por acá teníamos para comentarles qué es lo que está generalmente generando este flujo de calor. Bueno, entonces en este sitio, en general, en el área, tenemos algunos seamounts de mayor tamaño, que son los que están ingresando agua más fría al interior de la corteza, y en ese camino en el interior de la corteza se empieza a calentar con el calor de la corteza y sale en algunos montes más pequeños como el Dorado, en este que estamos visitando hoy, con un gradiente de temperatura mayor. Entonces eso es lo que estamos viendo, es un mecanismo y un mecanismo, digamos, hidrogeológico, donde en ciertos lugares y en ciertos seamounts o montes submarinos es mucho mayor, son mayores y captan, ingresan, digamos, más agua fría, y en otros sitios salen esta agua, digamos, pero de manera más caliente. Entonces, esto es lo que principalmente atrae a estos organismos, ¿verdad? Todos los que vemos acá, esta comunidad de organismos que tenemos acá, como lo que son los pulpos, lo que son Vemos casi siempre asociados estos camarones, también lo que son poliquetos. También hemos visto bastantes, o lo que son octocorales, bastantes esponjas, y todo esto, digamos, sería también parte de estos objetivos de volver hoy acá y poder entonces poderlos muestrear. Another thing that is interesting here is to look at the benthic community. The benthic refers to the organisms that are attached to the rock, like the corals, sponges, polychaetes. So this is, in those places, we have seen a community of these organisms. Also, as we can see, another organisms that are not benthic, but they are most of the time associated, like the shrimps that you are looking at now, close to the octopus, and also some crabs. And sometimes we have seen some special fishes that is also important to record. Aquí estamos, o se tratará de tomar una muestra de lo que es este flujo de calor con estos aparatos que están en este momento sacando acá los pilotos. Y lo que trata entonces es de sacar un poco de agua caliente, y después se analiza lo que son los microbios asociados, las bacterias. Y geoquímicamente también pueden ser analizados.
Looks like it's a little slack. Drop your arm down and see if it slips. I'm afraid to grab it anymore. You need that little much more, just a little.
Este sitio es impresionante. Vean la cantidad de pulpos que hay anidando sus huevos, aprovechando este calor y este espacio también limpio de lo que son las rocas. El agua al salir, entonces limpia la superficie de las rocas, cosa que también las pulpas aprovechan para tener pues su lugar de anidación limpio. Y ahí pues entonces anidan durante el tiempo de anidación, que ese no se conoce hasta el momento.
Sorry, which ledge was that?
So you see the octopus with the eggs? It's right up above her. I know where your shadow is.
Aprovecho para comentarles que en esta expedición tenemos diferentes expertos en distintas áreas, en un equipo multidisciplinario. La mayoría de personas investigadores que están acá son mujeres, entonces por lo tanto para nosotras ha sido una expedición bastante importante desde ese punto de vista, ¿verdad? Como es liderada por una científica también, esta expedición, por la doctora Beth Orcutt de Geological Institute. Y bueno, somos parte de esto ha sido importante, ¿verdad? Esa proyección que tiene esta expedición liderada principalmente por mujeres. Y pues en distintas áreas tenemos investigadores desde el punto de vista, ¡ay, sí sirvió! Qué bien, sirvió bien. Se alegran aquí un poco de emoción en el cuarto de control, ya que sí funcionó la toma de la muestra. A veces es complicada de tomar.
Okay, I figured it out. It's just timing it that little tiny bit more. Yeah, the plate on plate. It's too sticky. It's tough.
Here in the control room, they are very happy because the squeezer sample worked. Sometimes it's hard to difficult these kind of samples. So they're very happy about it.
So the one in the top port is another squeeze kind, and then the one in the back starboard is one that J-Rod made that we're trying to pull it. So I don't know how that's gonna work. We'll try that one last.
It's gonna work also.
I'm sure it's gonna work.
Yeah. Oh, you're gonna like hold it with one hand?
Yeah.
Okay, I see.
El equipo de investigadores, como le comentaba, a bordo, tenemos principalmente personas biólogas, pero también, como en mi caso, que estudié o mi formación ha sido principalmente geológica. El análisis principal desde mi punto de vista ha sido trabajar los sedimentos. También tomamos muestras de algunas rocas que tanto se van a colectar, fueron colectadas y van a ser preservadas en lo que es la Escuela Centroamericana de Geología, en la colección de rocas, pero también serán llevadas a análisis y lo que es conservarlas en la USGS. Además, tenemos investigadores que analizan el punto de vista microbiológico. Tenemos colaboración con otros investigadores que en este momento tal vez no estén acá, pero que están en un continente, como le decimos, desde otros institutos también colaborando con nosotros y analizando, aprovechando estas muestras no solo para los estudios de nosotros individualmente, pero también los estudios de estos investigadores. Les vamos a llevar entonces también parte de estas muestras y compartir las muestras para que analicen otro tipo que realicen otro tipo de análisis, ¿verdad? Puede ser interesante estas muestras desde otros puntos de vista. Por lo tanto, este equipo, estas muestras son aprovechadas al máximo, ¿verdad? No crean que se toman por tomar, o sea, son muestras que valen muchísimo y que se están aprovechando desde diferentes puntos de vista científicos. all the samples that we are taking here are also or contribute to do some studies that will be made for the scientists on board, but also some of these samples are also shared with other scientists in other institutes, and that we can different types of collaboration. For example, from the geological part, the part that I know, our samples from sediments will be analyzed. The mineralogy, we will analyze also the physical properties. We are also analyzing the biogenic content, the age of the sediments, using microfossils, but also we can do, depending of the type of sediments and the age, that we can do also some radiocarbon analysis. But there is here on board, there are different types of scientists that work in different goals, with different goals. For example, microbiologists, all the biologists on board, and all these biologists have their experience in different type of animals of the deep sea. So that's also a very interesting part of this expedition.
That's okay.
Yep.
You can use it as a background sample. Before you put it away, can you just get a picture of it? Sure. Rob, I got a picture. You did? Okay, great. You want to put that back?
Aprovecho para comentarles también que la página, que sigan o que ingresen a las páginas o a la página principal del Schmidt Ocean para que puedan seguir los blogs que los científicos han estado realizando. También tenemos los blogs de los dos artistas que están a bordo con nosotros, tomando lo que son todas experiencias y desde un punto de vista, un paisaje, ¿verdad? Marino, y tomando todas estas ideas para poder realizar lo que son la inspiración para realizar pinturas. Así que los aprovecho para que puedan seguir el blog y lean las historias.
Monkey's fist is to activate it. Don't grab that. Yeah. I think this loop is just the same. That's the one for holding it.
Yeah.
Okay. Yes. That's my kind of work.
Sorry. Oh, Dorado. Hey, I did put a red line on that tube, so when it's pulled all the way, see that it's fully.
You're way behind me. Yeah, so that's fine. No worries.
Yeah, I think Tim started priming it with just a tiny little bit of water, which is a great for the sample quality because then we have to back calculate, but rather than not having a sample.
That's when, remember, you can just grab it as hard as you want. It's not going to.
Just try not to get that with rope.
Estamos tratando de tomar diferentes muestras de lo que es el agua esta termal. con estos aparatos que les llaman squeezers. Lo que hace es que al abrirse, o sea, trata de ingresar al vacío la muestra de agua y luego se cierra. Y a veces funciona, a veces no funciona tan bien, entonces aquí se escuchan a veces los investigadores detrás, ¿verdad? Que están contentos si funcionó o no funciona la muestra.
Yeah, you can bring the rail in, if you do, bring that rail in.
También vemos algunos, digamos, experimentos que se han dejado ahí con algunos otros tipos de materiales, ¿verdad? Que no son precisamente rocas, como por ejemplo este PVC que ven. Ahí se dejaron hace unos días que estuvimos acá y ver si, por ejemplo, los organismos, ver cómo reaccionan los organismos o cuál es la reacción de ellos ante estos materiales pues nuevos, ¿verdad? No serían rocas principalmente, como lo que ellos están principalmente acostumbrados, es lo que tienen a disponibilidad.
It pushed. Yep.
Les recuerdo que o les comento que estos rocas que vemos acá, que se limpian a lo que es se limpia a la salida de lo que es el flujo de calor. Y estas superficies limpias también son aprovechadas por lo que son los pulpos para anidar. Bueno, aquí vamos a intentar hacer el muestreo. Ujú, ujú.
That's where I stop there.
Okay. Go. Pull back in, just, yeah. Scooch, man. Pull out again, or?
Yeah.
Pull it back. Okay, I'm good. So I probably watched that porch where he shouldered out.
Yeah, I was watching it in HD2. It was fine.
Okay. Let's hold it for a little bit.
Equalize them.
Yeah.
I think instead of let go, just like start going towards it a touch. Seem to hold it.
Yeah, sometimes.
Okay, let go, or?
Yeah, go ahead, let go. Huh? Weird.
turn the tip towards us?
Este sector, el Dorado, ya ha sido visitado en expediciones anteriores y en esta expedición la hemos visitado 2 veces. Esta ha sido, para nosotros ha sido un sitio donde se han observado, por ejemplo, hemos podido responder algunas de las preguntas que se tenían o hipótesis de las que se venían, ¿verdad? Con esos propósitos. Y en este caso, por ejemplo, vimos bebés, hoy no hemos visto bebés todavía de pulpos, los crías, pero esto fue uno de los principales logros de esta expedición, ¿verdad? Ver que esos huevos de pulpos estaban maduros y habían pulpitos dentro y inclusive vimos lo que son los bebés, las crías y esto ha sido uno de los logros principales, además de seguir, ¿verdad? Haciendo mediciones de temperatura. La idea es poner también o han puesto algunas muestras o experimentos principalmente para hacer otro tipo de observaciones con respecto a materiales, y que no sean principalmente roca o poner algunas rocas distintas también ha sido parte de los objetivos en estos experimentos para ver cómo reacciona este tipo de experimentos a este ambiente. Ujú.
See what's going on.
That could definitely be why some of them don't fire too.
Oh, if they're just kind of neutral position is close.
And the spring isn't strong enough to do the T4 is strong, so it's gonna be able to pull it up no matter what.
Janet was describing their rate of moving up.
If it's blowing its nose.
El sitio del Dorado se encuentra a unos 150 kilómetros de la costa en el norte de... ¡Ay, un bebé! Justo. A baby. Yeah.
Hold on, I'm gonna move away. Is that
Yeah.
okay, Mike?
Oh,
Gonna lose it?
I,
There it
is
is.
there? Yeah.
Yeah, it's there in the background.
Bring the cathets up.
You can see the baby.
Bye-bye, baby.
He's swimming. Bye bye.
So he came out.
I was.
Right under the
Keep
male.
the camera up.
Yeah. Turn on the.
Back. He came out from below where the male is.
Bueno, aquí tenemos una vista bastante bonita de lo que es la superficie de las rocas, cómo está llena y aprovechada por todos estos organismos de octocorales, esponjas. Vean los colores, cómo varían. Eso es un sitio, no nos cansamos de ver, la verdad, es increíble. Tanto la diversidad como la abundancia de organismos. Y también es un sitio de los pocos, o de los, por decir, único en el mundo, que se ha visto este tipo de diversidad a esta profundidad. Estamos a 2.994 metros de profundidad.
Feel it a little close.
Bueno, justo estábamos hablando del bebé que no lo habíamos visto y tuvimos suerte, lo pudimos ver un poco rápido porque nadó bastante rápido, muy enérgico.
this? Yeah, we did that. Did that. Did that. Okay. Thanks.
Yeah, sure. That's interesting.
Come up right real quick. Our next operation is we're gonna grab the temperature probe out of Biobox 1B. That'll oil and roll shape.
Watch out. Yeah, I think it's just
Dígale a Beth. Porque ahorita están con lo de los osos
Sí, sí,
cuidan,
pero
¿no?
después.
Sí, claro. Si yo estoy a esos. A veces que se ven ahí. Sí, el rosado, eso. Es que
If you're just tuning in, but you maybe saw earlier dives this expedition, we are returned to Dorado Outcrop, which was the site of our first couple dives of this expedition. We're currently just shy of 3,000 meters below the surface. And for anyone who maybe this is your first time tuning in, you are watching live footage from the remotely operated vehicle SuBastian, a remotely operated submersible, like I said, about 3,000 meters below the surface of the ocean. And we're operating this from onboard research vessel Falkor 2 by way of a very long umbilical tether, which relays control from the pilots here on the ship down to the remotely operated vehicle. And it relays the video from the vehicle back up to us. And then we can relay it out to you through the wide web. If you are tuning in and you have any questions about the work being done here or about Falkor 2 and SuBastian, the operation of, feel free to type those in the chat, and we do try our best to get to those questions when we can.
you want me to hand you the grip or? Mine is lost and just bound in the tip end and okay. So we'll hold catch like moving. All right, gonna close the final box. Unfortunately. So we're putting this in the same sort of place.
Yeah.
This or below it.
Ideally, the one that's upside down would be like behind it with the tip facing towards the one with the eggs. If that makes sense.
I bet if you rotated the rope, it would like pitch it. Eventually. About it the right direction. Maybe not. Just turn. You wanted it above that with this head.
This is good. Because I don't just get it up there without like, getting on that really. So I can do exactly what you're doing.
Well, here that we have is a temperature logger. So we will leave it here until December. That is the next expedition. And it will take each 30 seconds the temperature, registering the temperature. Bueno, entonces aquí lo que tenemos, este es un medidor de temperatura que se va a un registro, lo que va a mantener es un registro continuo de la temperatura cada 30 segundos y se va a mantener así hasta aquí, en este sitio donde están intentando ponerlo, hasta diciembre, que sería la siguiente expedición, donde volveríamos a ver, ¿verdad? El registro se va a obtener y después se pueden ver todas esas variaciones de temperatura en este sitio. Se está eligiendo este sitio, pues es uno de los lugares que hemos medido la mayor temperatura o la mayor en este momento, en este lugar.
Might be hard to get the tip on that ledge. Yeah. You'd have to do a solid grab, but then letting it go would be kind of difficult. Yeah. That's a sweet site.
Aprovecho para contarles que en esta expedición hemos tenido o tenemos la participación de 8 costarricenses, en lo que son 7 científicos y un artista. Por lo tanto, creo que es una de las expediciones que hemos tenido mayor participación de nacionales y también involucrados en lo que es la investigación, puesto que a veces hay expediciones de este tipo que se vienen a nuestros países, ¿verdad? Vienen a nuestros países, pero no hay tanta participación o la participación es nula. Por lo tanto, esto ha sido para nosotros es un gran logro y también poder analizar nuestras propias muestras en los laboratorios y en las universidades respectivas. Tenemos un investigador de la Universidad Nacional y 6 investigadores de la Universidad de Costa Rica.
Well, just to the right. Yeah, to the right of the arm. Right up. Okay, well, there's many of them. Got a porch out. Yeah. You can grab all 5 at once if you want. Or you can do them all kind of different. Altogether, they weigh 45 pounds. Before you set them,
Good.
oops, that's fine, don't worry about it. Nothing happens. If you could pull that bundle over to the starboard side just a little bit, I want to view what is on this ledge. It's like a little fish. No, you're fine.
Oh yeah, we saw one of these under a rock one time.
Yeah.
Beatriz told me that if we look at small fish, they found a species in the infancy in Jaco, and now they was crying, so she told me if we look one, we can catch one.
We didn't get a permit for fish. fish. Yeah. Okay.
There it is.
Hopefully, the fish will relocate when they want to put all the stuff there.
Bueno, estamos viendo aquí un pez, este pez blanco se ha avistado, yo creo que no sé si es la primera vez o pocas veces en este sitio, y también se había encontrado en algo similar, no exactamente esta misma especie, en lo que son las emanaciones de metano en lo que es el Pacífico Central. Bueno, aquí también parte de los objetivos es dejar algunas trampas para lo que es la actividad microbiana. Estas trampas tienen algunos materiales distintos a los que están acá. Entonces se van a hacer algunos experimentos, cómo es esa reacción de esos microbios a este tipo de ambiente.
Yeah, is locked for deployments around?
Yes. Nice. The fish is giving us some real steam.
Yeah.
Oops.
Maybe one at a time. I know it's time consuming, but it's cool.
Are those the experiments of Gus, right? Dentro de estos experimentos, el compañero que estuvo antes que mí acá acá narrando es el doctor Gustavo Ramírez, que tiene parte de su investigación es dejar estas trampas microbianas con distintos materiales adentro y a que se dejarán para luego analizar y recogerlos en diciembre, a ver qué microbios y cómo han reaccionado a los diferentes materiales que están ahí en esas trampas. The same as you think so?
Yeah. I don't know if it's the same species because they just describe it from the 1960s. And this is, the color is different.
Okay.
So it might be something new. Yeah, in the US, it's anything that's a vertebrate is rules of vertebrates. So that's why I just wanted to double check.
limestone, and an inert, which is like borosilicate B10.
Okay. Yes. Oh, so to explain a little bit about what we are living these experiments here, those are different kind of materials in one of the samples. So the idea is to the experiments is to leave a different kind of materials there. Then there are 5 different type of materials, and one is glass, and another is steel, and another one is rhodochrosite, that is a carbonate, magnesium carbonate, and also there is different type of basalts too. One of like fresh lava, and the other one is a old lava. And so the scientists want to look at the microbes. What is the reaction, or what is the community of the microbes after some month after. So we will collect them again in December to see what was the community of the microbes, and to look at the different materials. This is an experiment of our colleague, Gustavo Ramirez, from the California State University. Para comentarles un poco sobre estos experimentos que se están dejando acá, son diferentes materiales, son 5 tipos. Unos son diferentes tipos de lava, una lava lo que es una lava reciente, una lava de hace 23 millones de años, otra lava, o lo que es vidrio. En otra tenemos acero, y en otra lo que es un mineral que se llama rhodochrosita, que es un carbonato de magnesio, y se dejarán acá para observar lo que es la reacción, ¿verdad?, que tienen los microbios a estos diferentes componentes que se están dejando en estas rocas y en este ambiente. Que se dejarán acá hasta diciembre, que vendríamos a recoger las muestras y luego serán analizadas por el doctor Gustavo. Les recuerdo que en este sitio, estos son montañas submarinas, lo que llamamos montículos submarinos. Estos son de pequeño tamaño, entonces son un tipo de montículos de lava. Estas lavas vienen desde lo que es la dorsal del Pacífico, provenientes. Estas han sido datadas o tienen una edad de aproximadamente 23 millones de años. Todas estas costras que se le pueden ver a veces unos colores, ¿verdad? Esos a veces son oxidación como óxidos de hierro. También en algunas partes se ven como más verdosas. Estos son este material que utilizan los pulpos para pegar los huevitos en la lava. Y efectivamente estos son un tipo de lavas volcánicas, se llaman basaltos. Estos basaltos, al salir de la dorsal, ¿verdad?, de esta gran fractura, por decirlo así, sale el magma, y al entrar en contacto con el agua, ese magma, con el agua de mar, entonces se forman unas bolas redondas, por decirlo de alguna manera, que le llamamos almohadillas. Y porque tienen forma de almohada. Entonces, este es lo que vemos un poco acá, estas formas un poco redondeadas, respondiendo a lo que son los pillow lavas o lavas en almohadilla.
Although, before you set that down, I apologize. There's a tiny little fish once we're done with this.
Yeah, that little thing.
The outcrop that we have here is called, as you know, Dorado Outcrop, but is principally consists principally of basalt, lava basalt, and these basalts, when are coming out from the, or they are originated in a type of mid, we call mid ocean ridge, and these one are coming from the East Pacific rise, and they are probably when they were doing some dating, and the age is around 23 million years ago, and when these lavas are coming out from the ridge, so they form this like type of balls that we call pillows, because they look like pillow, like a pillow. So they are made from the basalt, and they form this spherical type of spherical form that is called a pillow lava.
Beautiful. Thank you.
Okay, so before that little fishy gets away, I think we want to try to get a slurp sampler out. So that we can try to make that collection, and then if we're gonna have a slurp out anyway, we'll also get some shrimp and another section sample.
Vamos a colectar un pez, pues el pez que vea, que de hecho recolectamos porque se encontraron una especie muy consistente de
Sorry, Joe. Offshore.
aquí algunos. Ya dejamos las muestras de
There's some coral over here that they wanted to look at.
y deploy microtraps, esas también. Entonces ahora vamos a tratar de hacer algunas recolectas, pienso. Este zoom lo quieren ellos.
Can you take some zooms, Rob?
Y
Where's our chat?
me imagino que también un listing. Ajá. Pero ya todo esto se ha hecho. Sí, y hacer algunas colectas y ese pez que quiere. Sí.
Yeah. We'll get this kind of wider shot, and then we'll try to also get some closer to the zoom. The polyps, but it's going to be hard with this shimmering water.
Ujú. Bueno, acá esto es lo que estamos viendo, es un octocoral. El interés de la investigadora que estudia corales acá es hacer algunas buenas tomas de estos corales para poder identificar, y por lo tanto estamos haciendo hacemos unos acercamientos al coral, a los pólipos. Vean que tiene 8 tentáculos, por lo tanto son octocorales. Ah, bueno, ya se fueron. Entonces ahora los dejo con el otro investigador.
Looks like we've got handover coming.
Los dejo con el investigador Sergio Cambronero del Departamento de Física de la Universidad de Costa Rica. So thank you everyone for being here this hour with me. So I will leave you with Sergio Cambronero from the Physical Department of the National University here in Costa Rica. Bye.
Well, it's a good time to catch anyone who may have just joined us. You're watching live footage from about 3,000 meters below the surface of the Pacific Ocean off of Costa Rica. This footage is coming to you by way of research vessel Falcor 2, but the footage itself is being captured by a robotic submarine, a remotely operated vehicle, or ROV, SuBastian, down at the very end of a long tether umbilical. And our pilots are currently coordinating two manipulator arms here to position a suction sampler as we prepare to hopefully collect another specimen.
Oh, the suction samplers.
Muy buenas tardes y muchas gracias a mi colega María Sandoval de la Universidad de Costa Rica. Mi nombre es Sergio Cambronero, soy profesor y investigador en la Universidad Nacional y los voy a estar acompañando por la próxima hora en este buceo que estamos en El Dorado. Good afternoon, everyone. Good morning, wherever you're tuning in. My name is Sergio Cambronero, and I will be joining you for the next hour while we finish our sampling at the Dorado Outcrop.
We do have a few more hours here, so. Sergio. Sorry. Also, in case you are just tuning in for the first time on this particular dive, what you see in front of you are in fact octopuses, many of them, and although they're kind of laying out in these weird positions, they are actively brooding. So many of these we believe have eggs underneath them. We've actually seen eggs, including eggs with eye spots, and we have seen hatchlings as well. And so scientists are interested in, one, what it is that makes this area so special for the octopus, but two, there's venting going on here, likely related. And so we are leaving behind many experiments and collecting as many samples as we can. And I would encourage everyone to check out the new blog posts over at schmidtocean.org. We've got a couple posts up about this expedition from the scientists, and they are fascinating reads.
Como lo explicaba nuestro compañero del Schmidt Ocean Institute, Alex, parte de los pulpos que estamos viendo en las imágenes que tienen en pantalla son pulpos que están vivos, a pesar de que se ve que tienen poca actividad o poco movimiento. Y estos pulpos que están, por decirlo así, como vueltos al revés, son pulpos, pulpas principalmente, que están incubando sus huevos. Estas pulpas, lo que se ha encontrado y parte de las observaciones que hemos hecho es que se agregan alrededor de estos sitios donde emana agua caliente de la corteza oceánica. Y justamente pues también parte del objetivo de esta expedición es entender los mecanismos geofísicos asociados a la emanación de este fluido más caliente. So thank you everyone who's tuning in and leaving your comments and questions. So yes, they're alive, as Alex said, and they are brooding their eggs on this pretty particular position. Also, you can see some of them, like the one you have on the lower left corner, doing almost like a crazy dancing, and that's a movement that they do to clean the surroundings around their eggs and also to ventilate them. So you can see them right now very close to the center. So yes, pretty particular behavior that we are observing right now.
So in the meantime, we can get some of the shrimp. And we'll see if he comes back up. You're an expert at this now. Just did that last time.
So this is our 9th dive of our Octopus Odyssey Expedition. We're currently at Dorado Outcrop, which is offshore the Pacific coast of Costa Rica. And on this dive, we have particularly been positioned on this same spot, which is the venting site that we have located and identified of this particular area that is called, again, Dorado. And we have been taking several samples in here. So we just have deployed many instruments that will allow us to know more about the water chemistry, to know more about the microbes that live here associated with this warm fluid. And we will soon be trying to collect more biological samples, not particularly octopus, but other associated organisms such as the shrimps that you see on screen, that they're usually associated with this warm fluid, and also some fishes that we have seen before that we would like to get on board. Also a good time to remind ourselves that all the samplings and everything that's happening in this expedition has gone through strict permitting processes with the Costa Rican authorities, and everything that is being collected of the animals' biological samples will be deposited in the Zoology Museum of Universidad de Costa Rica, not only for us to be able to do our further analysis, but also for international colleagues that might be interested to have the option to have access to these samples. So thanks everyone for leaving your thoughts and questions. Please keep them coming. We will definitely be reading them, and also would like to know where you're tuning in. Por favor, déjenos sus comentarios y sus preguntas. Si nos están sintonizando desde Costa Rica o otras partes de habla hispana. Uno de los objetivos principales del Schmidt Ocean Institute no solamente es la investigación en la ciencia, sino que también tener un impacto en la comunidad en general. Así que para nosotros es muy importante saber de dónde se están conectando, de dónde nos están sintonizando y responder sus preguntas. Eso lo pueden hacer a través de los canales de YouTube o de Facebook del Schmidt Ocean Institute. So we have some reports from Australia. So we're glad that this is being watched all over the world to Australia, probably nighttime by now. So thanks everyone who are tuning in this live stream, our 9th dive of our Octopus Odyssey Expedition. Also, we have some reports from England as well. So this is one of the most amazing things that technology allows us. And this is the first time that we are exploring these seamounts in the North Pacific EEZ of Costa Rica. So this is a pretty big moment for us, Costa Rican scientists, not only to be able to get all these samples, but also to be able to broadcast this live to Costa Ricans and people from all over the world.
So before I see 2 in there. There should be 3, right?
What was that?
You did see 3. Okay. Yeah. We'll go for index.
We see some comments and questions in regards of how large is this aggregation of octopus right here. So this is part of what are we going to have some preliminary answers once we get the analysis of the videos. But I would dare to say that last time, because we were previously here in our initial dives, preliminary countings were up to 1,000, 2,000 octopus. So it's large aggregations for sure. But again, this will be confirmed thoroughly after colleagues on board will take a closer look at the video and the video transects that we have been able to get from this specific location. So that was a great question. Thanks again for everyone who's tuning in.
While we wait to see if the fish will reappear, those corals that we were looking at just a moment ago, we want to try to collect those. We're going to try first with a slurp. If that doesn't work, then we'll try to pick them up with a manila later. But I think we'll probably have to reposition no matter what. Do you agree?
Oh, that's them up there?
No, not that far. Never further down.
Remember, I repositioned, so it should be further right.
Okay.
I think that they better try this.
So before you try, before you get closer to slurp on, you want to get some tight shots. Yeah, that's good. Just hold it there for a moment.
So what we're seeing here is a type of octocoral, and you can tell by the number of tentacles on the polyps. So corals are colonial organisms. So each one of those 8 tentacles features is actually one little animal that live in a colonial setting. So these corals are different from the ones that we have in shallow waters in many different things, but one feature is that the ones that we have in the shallow waters sometimes are called hexacoralia, and this only do have carbonate skeleton associated, while these other ones, their deepest counterparts are usually not related or not able to create this carbonate structures. So we have our Costa Rican expert in octocorals, Dr. Odalisca Brady from Universidad de Costa Rica, who is currently directing the sampling of this very beautiful specimens that we see here growing and thriving around these warmer waters.
Sí, es una zona probablemente es la familia Fauna que sí, pero no estoy segura. Tal vez ella se close-up después.
Se puede, okay. Can we get it closer?
That's full zoom.
That's full zoom. That's full zoom.
But we can try to get it, and then there's other ones around here. So once we reposition, we do other things, we can try and look at it again.
So Dr. Brady, who's our octocoral specialist on board, is actually really interested in this particular corals. What we know is they're from a group that's called Alcyonaceae, but also Dr. Brady recently described a new family of octocorals that was named Aquaombridae. So she has some, let's say, preliminary hypothesis that this might be associated with that family that was just recently described. So as you can imagine, these samples are pretty important for us to have a closer look once we get them back on board. There might be a new species, a new family. We don't know yet, but this is why it's so important to have these expeditions, and not only to get the samples, but also have this awesome footage, which is very high quality, that our colleagues from the Schmidt Ocean Institute provide us.
That's a good angle. It's decent. Better, but
So thank you everyone who's leaving their comments and questions in our chats in YouTube and Facebook. We're currently at 2,993 meters offshore the Pacific coast of Costa Rica, and we're gonna go ahead and try to sample this coral, which is, as you see, thriving around this hydrothermal venting site.
it's a great angle. It's great.
Get some highlights, please.
Okay. So we see some questions in regards to how old are these octopus when they breed. So it is a great question, and indeed, part of what we're trying to answer here is this type of questions. So currently, our expert on board, Dr. Janet Boyd, who is an octopus expert, just confirmed that there is no particular answer right now for that question, but we might get some answers eventually. So great questions, and thanks for tuning in while we sample this octocoral that seems very, very attached to that substrate. Yeah. So if this method doesn't work, we might go ahead and try a different technique probably grabbing the coral with the robotic arm of the SuBastian.
I think we can stop. So while we still have the stuff in our hands, we'll just look back to see if the
So
fish is there.
if you just paid attention to just what happened, that octocoral, when we tried to sample it, it retracted its polyps. So this is very common behavior that we see in these animals. So they usually have their polyps extended to catch the particles that are suspended in the water. But if they find any threat or a particular stress, they would retract their polyps.
We can go ahead and stone the start for now. While we're sitting here,
So thanks again for everyone who's leaving their comments. This octopus, we have a question. At what depth this octopus normally live at? This octopus are already identified as one genre, that is moose octopus with double U. And what the experts here on board have been telling us is that this might be even a new species of octopus. So imagine this is barely the one of the first sites that we're actually seeing this particular species. So as we know, if there is a new species, eventually it will be at the moment restricted at this particular depths, which are around 3,000 meters depth. There is another very similar octopus, also of the moose octopus genera, that lives in Californian waters. But again, that might be probably a different species, but also living at deep waters. So again, as the same way you're getting your questions, the scientists on board, we have lots of questions, and this is part of the exciting things about science and this type of research expeditions, that we are able to answer some of the questions, but sometimes we leave with more questions than answers. So that means that we are keep innovating and keep developing knowledge for whole humankind. Vamos a intentar colectar una muestra de un octocoral que estamos observando alrededor de estas emanaciones de agua hidrotermal. Hay un interés particular de la doctora Odalisca Bridi de la Universidad de Costa Rica de obtener esta muestra, ya que ella recientemente descubrió o describió más bien una nueva familia de octocorales, y existe la posibilidad y ella tiene la sospecha de que este pueda ser un coral que pertenezca a esta familia. Así que es algo muy interesante porque muchísimas de las cosas que vemos en estas expediciones pueden ser nuevos hallazgos para la ciencia. So thanks everyone. Keep leaving your comments and questions. We see some reports from Australia. Got a couple of folks in the chat from Australia. Also from, I think earlier it was mentioned from the US, from Costa Rica. So thank you everyone for joining. Also, we see some reports from Finland. And this is very important for us to know that our impact and the outreach efforts are actually being watched from all over the world. So what you see on screen now is the triggering of a specific sampling technique that is specifically to sample water, and these are called Niskin bottles. So as you see, the robotic arm triggers this specific whitish piece of plastic, and this closes the Niskin bottle, which captures, let's say, a parcel of water from this particular depth. So once we get the sample back on board, our scientists will be able to take a closer look at the not only the chemical composition of the water, but also the microbes that might thrive in this particular waters that are inhabited and associated with this brooding octopus. Así que un gran saludo a todas las personas que se conectan. Vemos que se conectan desde Finlandia, desde Chile. Tenemos personas de habla hispana conectándose de diferentes partes del mundo. Para nosotros es un gran honor poderles estar haciendo esta transmisión en vivo desde Costa Rica, mientras estamos viendo los diferentes muestreos que estamos haciendo en esta locación particular que se llama el afloramiento rocoso dorado.
Do you think that we can try to pick this up? Yeah, let's try to get that one.
You see the lasers? You see the right laser?
So we do see a comment in the YouTube channel. Just to clarify, this is our 9th dive, not necessarily the 9th expedition. So during one expedition, we are able to have several dives. So yeah, just to clarify, this is the 9th dive, and this is the first leg of this Octopus Odyssey expedition. We will be coming back hopefully the next December to keep doing our observations, but also to retrieve some of the experiments that we're leaving here at this particular site. There was initial and previous expeditions to these sites back in 2013. There were several ones around those years that were able to map and get temperature readings, also some initial preliminary observations of this octopus brooding behavior. But again, those were different expeditions, different objectives, and thanks to them and to those amazing efforts, some papers got published, and because of that, that was the initial reason for the interest on coming back again and have a further look and detailed observations of this site. And if you have not been following our dives, I will totally recommend you to go to the Schmidt Ocean Institute channels, whether YouTube or Facebook, because they have all the recordings from previous dives. So last dive, we were able to find and discover another octopus aggregation at a different site, which we also found hydrothermal venting. So this is already really exciting for us because we're not only collecting and understanding more about this particular site called Dorado, but also we're discovering similar habitats and ecosystems around these areas. And all these seamounts are part of, let's say, wider area that we call the northwestern seamounts off the Pacific coast of Costa Rica. So thanks everyone for leaving your comments. We see many great questions. Y un gran saludo a todas las personas que se conectan desde Costa Rica. Por aquí vemos que nos están sintonizando desde la zona norte de nuestro país, nuestra querida Guanacaste. Un saludo muy, muy caluroso y especial a la familia Lara en Coajiniquil. De verdad que para nosotros es un honor estar acá representando a todos los costarricenses que tenemos un gran amor por nuestros recursos marinos y nos da muchísima felicidad. Estoy seguro que no solo a mí, sino también a todos los ticos que estamos acá, ticos y ticas, ver que nos están siguiendo y siguiendo esta expedición desde sus casas. Y bueno, particularmente de nuevo un saludo muy especial a la familia Lara por allá en Cuaji. Espero pronto estar por allá. Pura vida. Un saludo a... También vemos colegas que se conectan desde Chile. Un poco más al sur de donde estamos ahorita, pero al fin y al cabo también conectados por las corrientes marinas. Así que saludos a nuestros colegas chilenos. Y bueno, si están escuchando esta transmisión en vivo, son en este momento las 4:30 p.m. hora de Costa Rica. Estamos aproximadamente 3.000 metros de profundidad en este sitio que le llamamos el afloramiento rocoso dorado, en donde podemos ver todos estos pulpas en su mayoría que están incubando sus huevos asociadas a agua caliente que emana de la corteza marina. So lots of comments and questions. This octopus are preliminary identified as moose octopus by our experts here on board, and there's a huge possibility that there might be a new species. So thank you for that question. And we also see a question that what are the PVC pipe that we put down here? So these are experiments to test different hypotheses, mainly to see if the octopus prefer hard substrate or if they're actually associated just with the warm water fluid. So as these PVC tubes that you see here, we have also deployed different shelters. And yes, so these are experiments and we will be picking them up later on. Usually when we have the possibility of visiting these remote places, we try to make the most out of it. So many different experiments are being carried along with the observations and the sampling. Again, because at least, and this is particular from Costa Rica, we don't have the capacity with our national vessels to go as deep. So this is a very special occasion for us to have the option to be on board the Falkor 2 vessel from the Schmidt Ocean Institute. So special thanks to them for making this possible.
We're gonna take a glamour shot.
We're currently carrying part of the geological sampling. We wanted that rock, but as you can see, sometimes plans do not go accordingly, but that doesn't mean that we're not able to get the samples. So we have a very skillful team here in the control room. that have masterized the whole maneuvering of this ROV. And I saw early in the YouTube channel that we had a question about if there is any delay between the commands that pilots give here in the control room and the response of the ROV. So from what we heard and what we have been able to see, this delay might exist, but is very, very minimum. So it's almost real-time response. And this is part of the amazing things that this ROV SuBastian offers to us.
Yeah, when you were going in, it was great, and you just caught fingers, and that's how you were like, let me get it. You know, like sometimes it's worth just actually 30 seconds to try to get fingers wiggled underneath it.
Un saludo de nuevo a todas las personas que se conectan desde Costa Rica y otras partes de habla hispana. Esperemos que estén disfrutando esta transmisión, tomándose un cafecito, un té. Y estamos viendo acá esa agua que se ve así como nubosa, es lo que le llamamos shimmering water, y eso es una señal de que es agua que está saliendo con una densidad diferente desde la corteza marina. También vemos que esos pulpos particularmente están asociados a ese flujo de agua caliente y, como pueden ver, están posicionados encima de una masa que pueden ver que es como pequeños cilindros blancos, y eso justamente son sus huevos. Así que todos estos que vemos aquí son hembras de pulpos o pulpas, si lo quieren ver de esa forma, que están incubando sus huevos y protegiéndolos. Son historias muy particulares, ya que estas pulpas a veces tardan años, más de 2, 3 años en incubar sus huevos, y dejan de comer, dejan de alimentarse para dedicarse exclusivamente a cuidar a sus pequeñuelos, sus futuros pequeñuelos. Y es una historia, la verdad, bastante conmovedora, porque ellas van perdiendo sus funciones, su masa muscular, y con el objetivo de poder cuidar sus huevos y mantenerlos en las mejores condiciones. Y pues una vez que sus huevos logran eclosionar, usualmente las hembras quedan en un estado de, por decirlo así, de senescencia, en donde se encuentran débiles y al final pues terminan muriendo. Así que es una historia muy conmovedora, pero que al mismo tiempo nos deja ver las grandes maravillas que suceden a miles de metros de profundidad en el océano, y que si no tuviéramos esta tecnología y esta oportunidad, pues no nos daríamos cuenta de que esto está pasando. Y particularmente es importante porque no solamente estamos avanzando en la ciencia y en entender más sobre estos pulpos y su fisiología, su comportamiento, pero también un gran objetivo que tenemos, y se los digo como costarricense, es que todo esto se traslade a los tomadores de decisiones para que ojalá en un futuro muy cercano hayan medidas de protección y de manejo sostenible para todos estos ecosistemas de aguas profundas. Aquí estamos intentando recuperar un instrumento que se estaba pues ahí de manera accidental, pero gracias al equipo que tenemos a bordo, a los pilotos, lo estamos intentando recuperar, porque procuramos de que todo lo que dejamos para hacer un experimento o para hacer alguna observación, procuramos recogerlo para no dejar ningún impacto humano en el fondo marino. Y asociado a esto, me gustaría hacer el comentario también de que hemos visto basura marina a estas profundidades en buceos anteriores. Así que es un recordatorio para tomar conciencia de que todo está conectado y muchísima de la basura que no tiene un tratamiento adecuado y un manejo adecuado en tierra, pues puede terminar hundiéndose hacia estas profundidades e impactando estos ecosistemas que de alguna u otra forma no han sido tan impactados por el ser humano. So we see how the octopus push away these shrimps that are usually very conveniently located. Currently on your screens, you're seeing the robotic arm of ROV SuBastian going forward to this particular substrate to get a rock sample that it is very important for us to assess the composition of this substrate, but also the history of this particular place. Also, part of the main objectives of the expedition is to know what type of microbes are associated and living in these rocks. So this is a very, very important part of the expedition because although we're not able to see it with our bare eyes, there are many, many microbes thriving and living in these deep waters and specifically in the ocean crust. So this is, again, one of the main objectives of this expedition. And this component is being directed by one of our two co-chief scientists, Dr. Beth Orcutt from the Bigelow Ocean Institute. And basically, what they want to know is to characterize and know the potential ecosystem service that we might obtain from these microbes living at these depths. Also, if you're paying attention, almost in the very center of your screen, actually no. What UR is being shown? Is that one or this?
Oh, I can kick over to this one. It's the top one.
The top one. Okay.
Yeah, but I'll kick over to this.
Okay. Cool. All right. Thanks. So as I was saying, if you see at the very center of the screen, you see a couple of green dots. These are lasers that are used to measure things that we see on video. And these dots are separated from each other 10 centimeters. So they give us a spatial scale that allow us to assess
Okay, let's try to reposition that temperature probe.
Yeah, I do agree. Unknown explorer for sure. So again, we're gonna go ahead and grab some samples that will allow us to know more about this place.
So, sorry, talk about this. Before we had it kind of right in front of that octopus. So I checked it out. Where I wanted it actually is going.
So we do have little pointer sticks and mission control here. So the scientists can reach out and point at the monitors that the pilots are piloting off of. And Beth was just sort of pointing where we're hoping to deploy this temperature probe.
Yeah. So thank you everyone who's leaving their comments and questions. Keep them coming. We are more than glad to read them here and try to give our best answer. We have many specialists on board that are very keen to answer your questions, but also take into consideration that most of the time, many of these questions do not have a particular answer. So this is what we're trying to do here, filling those knowledge gaps that we have. Muchas gracias a todas las personas que nos han dejado sus comentarios y sus preguntas. Si tienen alguna duda, algún saludo, lo que sea, por favor no tengan pena y nos lo pueden dejar por acá en los canales de YouTube o desde Facebook. Y aquí de verdad que hay un grupo de personas que estamos muy interesados en tener este contacto directo con las personas en tierra. Así que de verdad, si tienen alguna duda, no importa qué tan básica pueden creer que sea, muchísimas de estas preguntas también son muy interesantes para que nosotros logremos aprender. Así que por acá los vamos a estar leyendo mientras estamos en este buceo, nuestro 9º buceo de esta expedición. Y por si se han preguntado qué son estas, por ejemplo, estas tubos de PVC, que son como codos de PVC, ¿verdad? Y qué son este montón de, como decimos en Costa Rica, chunches, o qué son este montón de cosas que están aquí en el suelo marino. Muchos de ellos son instrumentos que se utilizan para hacer experimentos. En este caso, los que tienen a su derecha son experimentos de colonización, que básicamente lo que se quiere comprobar es si los pulpos responden a un hábitat o a una superficie dura y si logran asociarse a ellas. Entonces, bueno, estamos viendo de que tenemos un resultado muy interesante porque hay definitivamente un pulpo, una pulpa ahí metida en este tubo de PVC. Y los que ven a su izquierda son experimentos más dirigidos hacia la parte microbiológica. Y de nuevo, es para ver qué logra colonizar estos sustratos, pero que estos eventualmente se van a recoger en otro momento cuando regresemos en diciembre, si todo sale bien.
So I kind of got to just grab straight on the T handle, straight out the handle. Like this. I think, yeah, once you get in there a little bit, it'll
Entonces, parte de lo que vemos en este momento, de lo que pueden observar en sus pantallas, son un montón de pulpas incubando sus huevos que están en esa posición que es como una posición vuelta al revés. Y esto lo hacen para proteger con su manto los huevos que están pegados al sustrato, y que es muy curioso porque ellos utilizan un líquido, por decirlo así, una sustancia especial que ellos mismos producen, que se les conoce como cemento. Entonces este cemento lo utilizan para pegar sus huevos al sustrato y que de esa forma no se despegue. Y el pulpo que ven en la parte superior es un pulpo que probablemente sea un macho, que no vemos en postura de incubación. Y si pueden ver, de alguna u otra forma hay, y es lo que también hemos visto, hay alguna especie de dimorfismo sexual. ¿Qué quiere decir esto? De que hay diferencias morfológicas entre las hembras y los machos. Así que esto es parte de lo que estamos tratando de comprobar en esta expedición, pero preliminarmente nuestra experta a bordo nos ha podido confirmar de que al parecer sí existe esta diferencia que estamos hablando.
straight at it. No, just keep rotate so it's pointed away from us. Okay, now pitch. Excuse me, yaw left. So exactly, yes. This guy. Yeah, keep going, keep going, keep going. Okay, now shoulder down, get back in the camera. Yeah, now rotate some more. Pitch some more. Go the other way. Something like that. You're almost at it. So, yeah. So, look at that in that dark spot. And try and let that out.
So currently, what you see on your screens is basically one of our instruments that we use to measure the temperature. This case is a temperature logger that has the capability of taking a record or a time series of the temperature readings that we can find here in this shimmering water that comes out of the ocean crust. And as you can see, has some octopus associated. And this is, again, part of the main objectives of this expedition. We do see a comment about if there is any reason why venting will be more likely to take place at seamount peaks. This is a very, very good question. I am not a geophysicist nor a geologist, but part of what we've learned of talking with our colleagues is that usually you will find this venting associated with particular outcrops that are associated with bigger seamounts. So the hypothesis that is being tested is a new, recently described mechanism that is called the hydrothermal siphon. And the dynamic of this is pretty much that bigger seamounts are recharge sites in which the water infiltrates through the ocean crust, then travels through the crust itself, and then find its way out, mainly because of a pressure difference between the recharge and the discharge sites. So this discharge is usually associated with cracks that we find in the seafloor, as the ones that you can see here, and also these places are usually not covered by a layer of sediment. That is one thing that we find at the very base of the seamounts. Y vemos por acá un comentario de un colega y compatriota costarricense que trabajó en el Parque Nacional Isla del Coco. Así que un gran saludo para todas las personas que se conectan desde Costa Rica. De verdad que esperamos de todo corazón que estén disfrutando estas expediciones y estas transmisiones y que se sientan igual de afortunados que nosotros de poder presenciar esto, ya que estamos marcando historia. Se está marcando historia en Costa Rica porque es la primera vez que se exploran estos montes submarinos y que se obtiene video en vivo con esta calidad. Así que bueno, muy felices de ver todas las personas que se conectan y me comprometo a pasarle su saludo a don Jorge Cortés, quien es uno de los líderes de esta expedición y gran experto de las aguas y ecosistemas profundos de Costa Rica.
That rock right here. Yep. Just spin that clockwise until it's pointed down. What we're going to do is try to put the T straight in line with your jaw. You know what I mean? So just rotate it in your jaw. Grip a little bit? No. It should just rotate. All you can do is just pull it back and let it slip.
Watch out.
Okay, let's do it with the other minute. Let's get it safe a little higher.
Again, thanks for all your comments and questions. Please keep them coming. This is great for us because it allows us to know more about the impact that we have with this live streams. As we see part of our sampling taking place at around 3,000 meters depth offshore the Pacific coast of Costa Rica. It is almost 5 p.m. Costa Rica time. So we hope you're enjoying this live stream. Hopefully with some coffee or tea. I know that if people are tuning in from Costa Rica, probably they're enjoying some nice coffee. Me too.
Barely have that. It's just in the tip. But that actually made. Oh, believe it or not, when you go to like a.
So many of the questions that remain unanswered are what is the chemical composition of this shimmering water and how this varies along the year, along the seasons. So, this type of expeditions are very important to find answers to these questions and also to understand better why this octopus aggregate around these waters. We do know that there might be an association, of course, with the temperature, but also part of our questions is if they might find another chemical signatures from these waters outflowing of the ocean crust. On your screen, what we have, which is like a instrument in the shape of a T, is a temperature logger, so very appropriate. But yeah, this allows us to have a series of measurements of temperature, and this is important for us to know how it fluctuates. One amazing thing that previous studies have found here is that this fluid oscillates and varies along with the tide. So that means that as you see the tide going up and down and the coast, you can see also the effect of this tidal forcing deep down at almost 3,000 meters depth. So this is something really, really interesting and a fun fact, just to remind ourselves that everything in the ocean is connected. So part of we've been seeing in previous dives are these shrimps. These shrimps are usually living around these waters, but also they are conveniently positioned for any chance that they have to grab the eggs of the octopus. They will go ahead and take it. So, not to say that these are bad guys, they're only trying to find their food, but um, they're definitely very opportunistic animals. So you see that the octopus do take very serious their brooding functions and their brooding. So they usually are seen, as you can see here, pushing away the shrimps out of their eggs, like saying, "Get out of my way." So, not to blame them, I will do the same. But yeah. And the bigger crustacean that we have in the lower portion of your screen, it's a type of a squat lobster, probably a galathade. And again, it's not very as insisting as the shrimps, but definitely is aware of what's happening as we see that shrimp going out with a couple of eggs. So, yeah, they are definitely taking advantage of the whole situation. So many thanks to all the people tuning in from different parts of the world. My name is Sergio Cambronero. Just got that question in the comments, so thanks everyone. I am really honored and happy to be here and having the opportunity to join you in this live stream. I am a marine biologist from Costa Rica and have been working in physical oceanography for the last 8 years. And for me, this is a dream come true to be part of this expedition. So I'm really, really happy to be able to connect with folks from all over the world. So thanks everyone. People also call me Checho, so whatever you find most convenient. And yes, it's a pleasure for me to interact with you. And we see some comments from our colleagues in Costa Rica. So I will answer in English and Spanish. So they're asking at what distance we are from the coast and what's the name of the site of the seamount where we are located. So we're currently almost 150 kilometers, so almost 200 miles. 150 miles actually offshore the Nicoya Peninsula in Costa Rica. And the name of this seamount or this outcrop is Dorado. That means golden in English. And the summit, let's say, of this outcrop is at around 3,000 meters, actually, where we are right now, very, very close to the summit. And the base is located around 200 meters below. So it's not a very tall feature. This is usually why it's called an outcrop and not necessarily a seamount. And well, this has been an ongoing conversation with our colleagues here on board of the actual naming of things. But usually, what would you name a seamount itself will be more, let's say, taller features as the ones that we have been exploring previously, such as Tengo Set Seamount or Fuente. Para el colega costarricense que nos dejó un par de preguntas en el chat, estamos aproximadamente como a 150, entre 120, 150 kilómetros de la costa, específicamente de la península de Nicoya. Este sitio donde estamos ahorita se llama el afloramiento rocoso o afloramiento basáltico Dorado. Y básicamente este sitio se puede considerar como un monte submarino, sí, pero parte de lo que hemos estado conversando es que debido a su poca altura, es más considerado un afloramiento o un... ahorita se me fue el nombre, un afloramiento o un montículo. Ese es el término que estaba buscando, un montículo, en contrario a otros sitios que son muchísimo más altos, como los otros sitios que hemos estado visitando, como Fuente y Tengo Set, que sí son, por ejemplo, muchísimo más altos. Entonces, para que se den una idea, ahorita estamos en la cima del afloramiento Dorado, que está aproximadamente, como ven en sus pantallas, en la esquina inferior izquierda, a 3.000 metros de profundidad, y la base de este sitio está aproximadamente como a 3.200. Entonces estamos hablando de 200 metros de alto. No es tan alto, pero hemos estado en otros sitios que la base está ubicada como a 1.000 metros de diferencia desde la cima. Yeah. Así que, bueno, son cosas muy interesantes que se encuentran por acá a estas profundidades, y aprovecho para darles las gracias y invitarlos a que sigan conectados a estas expediciones. Mi nombre es Sergio Cambronero y para mí ha sido un placer estar conectados con ustedes. Les voy a estar pasando la dirección del buceo a mi colega Natalie. Natalie es una colega que está pues ayudándonos acá y muy interesada también en estas investigaciones científicas. Bueno, al parecer me quedo por acá con ustedes y vamos a pasar a una parte muy, muy bonita de esta expedición y de este buceo. Mientras le doy la bienvenida a nuestro piloto, J-Rod. Vamos a una parte muy bonita de la expedición o de este buceo, que es a obtener imágenes que puedan ser utilizadas para transmitir la magia de este sitio que estamos viendo acá. Entonces ya terminamos gran parte de los muestreos y entonces nos vamos a enfocar en obtener imágenes bonitas y ángulos particulares para poder contar estas historias. Estas agregaciones de pulpos realmente no son comunes y eso es parte de la importancia de lo que estamos demostrándole al mundo, y es que anteriormente solo se conocían 2 sitios en donde se encontraban estas agregaciones de pulpos en el océano profundo. Una de ellas fue en California y la segunda era esta en el Dorado, que es donde estamos ahorita. Y justo en esta expedición, en nuestros buceos anteriores, logramos descubrir la tercera agregación de pulpos. Así que es algo histórico lo que está pasando, no solo para Costa Rica, sino que para el mundo entero, ya que estamos pues comprobando de que nuestro patrimonio profundo en Costa Rica contiene o tiene ecosistemas muy, muy especiales y únicos en el mundo. Así que muchísimas gracias por ese comentario. And we are just finishing our samplings. Actually, the majority of our samples have been taken, the biological, the geological, and the hydrographic samples. And we're going into a really nice part of this dive in which we're going to focus mainly on getting awesome footage that will work not only to know more about the anatomy and the behavior of these animals, but also to showcase the importance and the wonders that we can find at this ocean depths. So bear with us because you're about to witness very, very special imagery. And again, thanks to our awesome pilots here in the control room from the Schmidt Ocean Institute team. So I'm very humbled by all the folks leaving your comments in the YouTube and Facebook channel. For me, it is again such a pleasure to be able to have this opportunity to interact with you live. So thank you very much, and I really, really appreciate your company here, your virtual company. So as we were saying, we are focusing right now on getting awesome footage, awesome close-ups that will allow us to tell more in detail and to show the wonders that we can find here at these steps. So you can see all the textures of the skin of the octopus, the suckers. These octopus are characterized by having 2 rows of suckers. And at the very bottom or behind them, you can see their eggs popping, those white little, almost transparent round things that you can see below the arm of the octopus. So while we watch these mesmerizing scenes coming live from the deep of the Pacific coast of Costa Rica, I will answer some of the questions that we've been getting through our Facebook and YouTube channels in Spanish. Y tenemos una pregunta particular de si esto es una roca porosa, si hay una fractura por donde sale esta agua caliente. De nuevo, yo no soy geólogo, no soy geofísico, pero gran parte de la valiosa y grandísima oportunidad que no solamente he tenido yo, sino que también otros colegas, es aprender de los expertos que están acá a bordo, expertos y expertas de diferentes partes del mundo. Y entonces lo que hemos estado aprendiendo y lo que hemos visto es que se está comprobando un mecanismo geofísico relativamente nuevo para la ciencia que se le llama el sifón hidrotermal. Y básicamente este mecanismo ha sido visto solamente en otro sitio en el mundo, particularmente un sitio que se llama Baby Bear, si no me equivoco, en Canadá. Y este es un mecanismo que consiste en un fluido o un flujo de agua que se origina en sitios donde hay alta presión y en donde el agua se recarga ahí o se infiltra y luego viaja por la corteza marina hasta salir en otro lugar en donde hay baja presión. Entonces, como sabemos, si uno tiene un gradiente de presión, lo que se tiende a hacer es a buscar el equilibrio, ¿verdad? Del fluido lo que tiende es a buscar el equilibrio. Entonces viaja de donde hay mayor presión a donde hay menor presión. Ahora, ¿qué quiere decir esto en palabras simples? Lo que estamos viendo es que los montes submarinos sirven como sitios de recarga, o sea, donde se infiltra el agua a través de la corteza de estos montes submarinos, tal y como sucede en tierra, en donde el agua se infiltra por las montañas y esa agua se infiltra por el monte submarino, viaja por la corteza oceánica y mientras viaja en esa corteza oceánica va experimentando un intercambio de calor con el subsuelo marino. Esto lo que hace es que calienta esta agua hasta que eventualmente encuentra un sitio a través de estas grietas que se encuentran, por ejemplo, en el Dorado, que es donde estamos ahorita, y que no particularmente son fallas, sino que muchas veces son sitios que están desprovistos de sedimentos o son sitios en donde, pues, debido a su naturaleza geológica, se crearon estas formaciones que tienen diferentes espaciamientos entre ellas y entonces el agua, pues, por cuestión, digamos, de las leyes de la física y de la dinámica de fluidos, busca salir por estas grietas y es donde justamente vemos los pulpos. El agua que sale de estos sitios aproximadamente sale entre 2 a 10 grados centígrados más caliente que las aguas circundantes. Entonces, espero que esta explicación haya sido de utilidad para entender un poquito mejor. Y como les comentaba, es un mecanismo que solamente se ha descrito en otra parte del mundo. O sea, que tenemos un gran valor, por decirlo así, a nivel de conocimiento en Costa Rica, en nuestro patrimonio profundo. Y esto nos hace únicos, ya que, pues, si no se ha comprobado en otras zonas, es importantísimo explorar y entender estos sitios. Mientras vemos estas imágenes, Qué increíble lo que estamos viendo en este momento. Yo la verdad estoy anonadado. Increíble. Wow. Wow. Discúlpenme por, por mis gestos y I'm sorry for my sounds and gestures, but it is just too beautiful and mesmerizing um, to watch this octopus. From what we know, this might be a male octopus because, in contrast to what you see from the other ones that are, let's say, in a different position from the upside down, um, those are females brooding their eggs, and this might be actually a male that is associated with these females around. So beautiful imagery we were seeing here. This is a moose octopus genre, so it's a, let's say, a particular octopus that's the scientific name, and there are high possibilities that this might be a new species. So, for those who are just tuning in, we are currently in our ninth dive of this expedition offshore the Pacific coast of Costa Rica. This dive has been special because we have remained in this particular site that is called Dorado Outcrop and trying to get more observations and deploying more experiments in this place that we already know has this aggregations of brooding octopus. Right now, this is a very special part of our dive because we have finalized almost all of our sampling and we are focusing right now on just getting awesome footage and awesome imagery as the one you can see here. Sometimes we have other priorities such as deploying experiments, getting samples, exploring, mapping, doing video transits. But right now, we're entirely dedicating this next hours to obtain awesome imagery as the one you can see here that will allow us not only to know more about the physiology, the anatomy of these animals, but also to showcase and show the world how mesmerizing and how beautiful are these creatures down here. So we have a question in our YouTube channel about the shimmering. So indeed, this is a particular site that there is lots of evidence of shimmering of or water coming out of the ocean crust with higher temperatures. So just to clarify, the temperature reading you're seeing on your screen is from the actual sensor on the ROV that um, it is not specifically reading the shimmering water. Earlier, I'm sorry, we had the opportunity to get a specific measurement of this water outflow, and the readings were as high as 10 degrees. So yeah, this is relatively very warm water in relation to what you find in the surroundings. We saw other sites in our previous dives in which we found shimmering, but not as warm as this one. So every place, every spot that we have the chance is a piece of this big puzzle that pretty much can tell us more about the dynamics that we find at this depths deep down in the Pacific of Costa Rica. So this octopus that we're seeing here with its arms extended, usually, and what our experts have been telling us is that they have chemical sensors, for, to put it that way, that they have in their arms. So they're extending to sense the environment and to get a glimpse of what's happening. So as you can see, there are lots of flows coming not only from the crust, but there are also ocean currents taken at this particular place that also can vary and bring different water masses. This is something that we are really interested, but for the moment, we're not able to answer those questions because we might need another equipments to answer them. For example, what is the current velocity here? Where is the direction? How this changed throughout the day? How this changed throughout the year? We don't know that yet, but it's part of the questions that we will keep, hopefully, for future expeditions. So what you see on screen are the brooding octopus. The majority of them that you see in this position, like upside down, they are females brooding their eggs. This is probably a male, and what you see on the background is a glass sponge, which is a very interesting animal. The sponges have been shown to be one of the most primitive animals, and um, they are a really important part of the ecosystem of the deep sea. Also, we find some hydroids and octocorals, also the ones that you see on the back that are like whitish tubes are usually polychaete tubes. So if you pay attention, you can see evidence of all different types of life forms that show also the complexity of these ecosystems and also the diversity. The one that you see at the bottom is a sea star. From this angle might look like a solasterid sea star, but I'm not sure. And the other one, this is a sponge. This is more, let's say, colorful than the other one. And we actually were able to get a sample of these sponges back in our previous dives. So we will be working with these samples to try and find out whether they're different species through genetics and genomics. So right now, as I was saying, we're focusing on getting awesome imagery and footage, different close-ups of the different species. So bear with us because we are just getting incredible and mesmerizing video live from the depths of the Pacific coast of Costa Rica.
Ya, pues, o sea, yo ahorita estoy bien. Sí, ya, está bien. Ah, bueno, ok. Entonces, si quiere, yo me voy a las 6. Yo lo paso a usted. Está mal, en verdad. Sí, ahora, ok. Ahorita. Estoy matizado. Sí, la misma. Sí, sí, ok, sí. Sí, sí, está bien, está bien. Ok, ok, está bien. Ajá, son hidroides. Ajá. Ok.
So this, what you are seeing on your screens, is a close-up of one of the female breeding octopus. And this is evidence, this movement of their respiration. So this footage is not only useful to showcase the beauty of these animals, but also to know more about their physiology and their behavior. So as you see, they're breathing. We can, for example, estimate their respiration rates out of this footage and see if they use this siphon to for another particular functions. We've been seeing that sometimes they use it to like clean some of the debris that is around. And I was fortunate enough to work some of the samples that we have been getting, along with and helping the expert that we have on board, Dr. Janet Boyd. And one of the most amazing things that I found, or that we found, that she showed me while we were taking the photographs, was some sort of reproductive organ that came out of this particular hole of this siphon that the octopus have. So this is actually the place in which they have their sexual organs and in which they mate with their male counterparts. So it was kind of like a transparent tube coming out of this siphon. It was long enough. So that was pretty awesome to watch. So we do see some comments in the YouTube and Facebook channel. So we see a lot of excitement, not only around the octopus and the awesome imagery that we are getting, this very good close-ups, but also we see some excitement around the sponges that we saw earlier. We had a question if that we're seeing is the siphon. So yes, I can confirm that's the siphon. Right, Dan? That's the siphon, right? So yeah, I was just double checking that I was not misleading you, but yes, that's definitely the siphon. And yeah, we do agree that it is very, very incredible to see how they respirate and how they use this for different functions. And as I was telling you before, we saw earlier when we got the samples back on board, the reproductive organ coming out of this particular orifice. Para los personas que nos están viendo desde Costa Rica y otros lados de habla hispana, estábamos comentándoles acá en inglés que estamos en una parte muy bonita de este buceo en donde estamos básicamente dedicándonos a obtener imágenes cautivadoras y que nos permitan entender más de la anatomía, de la fisiología y los comportamientos de estos animales, pero que también principalmente son imágenes que nos van a permitir mostrarle al mundo lo importante de estos sitios. Y eso es a través de esto, de capturar los comportamientos en vivo, en detalle, y poder ejemplificar la belleza de todos estos ecosistemas que tenemos en las aguas profundas. Comentábamos anteriormente ese especie de agujero que se veía que tiene el pulpo. Eso es lo que se le llama el sifón del pulpo. Y básicamente a través de una serie de observaciones, uno, por ejemplo, puede tener una estimación de la tasa de respiración de los pulpos y también ver si utilizan el sifón para otras funciones. Les comentaba que tuve la oportunidad de ayudarle a la doctora Janet Boyd, que es la experta en pulpos que tenemos aquí a bordo. Tuve la oportunidad de ayudarle a procesar algunas de estas muestras que hemos estado obteniendo, y algo muy particular que me llamó la atención y que yo nunca había visto fue que ellas tienen su órgano reproductor asociado a este sifón y a través de ahí es que lo sacan. Y es como un tubo transparente en el que entonces utilizan para copular con sus contrapartes masculinas. Así que esto que estamos viendo aquí en vivo, este comportamiento que vemos que es casi como un baile, es una forma de los pulpos de limpiar sus huevos y de oxigenarlos. Así que pues no hay mucho que decir más que contemplar estas bellezas que tenemos la oportunidad de ver en vivo desde las profundidades de Costa Rica. Vemos una pregunta en el canal de YouTube, que cuál es la diferencia de la salinidad y el oxígeno con respecto a la superficie. Es una muy, muy buena pregunta. El tiempo que tengo ahorita es limitado, entonces, porque ahorita otros colegas me van a continuar la transmisión, pero de manera muy resumida, la diferencia que encontramos entre la superficie y estas profundidades es algo pues muy leve, por decirlo así, pero que sí nos dice muchísimo de la diferencia de condiciones. Por ejemplo, en las aguas superficiales, en la capa más superficial del océano, donde hay una interacción con la atmósfera, tenemos que las concentraciones de oxígeno pueden ser de 200 micromolar a 250 micromolar, y eso es porque está en constante intercambio gaseoso con la atmósfera. Como ven aquí, las concentraciones de oxígeno que obtienen en la pantalla son 112 micromolar, Entonces son aguas oxigenadas, sí, pero que tienen menor concentración de oxígeno. Ahora, eso no quiere decir que sean aguas, digamos, con concentraciones bajas, que no pueda vivir la vida y justamente por eso es que vemos muchos animales. Pero nada más quería hacer hincapié a que entre la superficie y el fondo pasan muchísimas cosas. Y entonces es una invitación a ustedes también como para que pensemos cuando hablamos del océano en tres dimensiones y que particularmente en estos sitios encima de este fondo marino tenemos una columna de agua de 3.000 metros de profundidad que tiene una zona de mínimo oxígeno que se ubica usualmente entre 200 a 800 metros de profundidad, en donde las concentraciones bajan drásticamente, inclusive hasta cero.
And for all the animal stuff, we want to make sure we can take shots.
Jerry. Your mom said hi.
Oh.
That's so cool, man.
Okay, you try it. No, no. I meant just here. Yeah, so we're just gonna do animal collections now until the end of the day.
Okay. Okay, cool. So I'm just oh, you're up. Vemos unos comentarios en el canal de YouTube, especialmente de algunos colegas interesados en obtener observaciones de otros organismos. Voy a intentar lo más posible que lo podamos hacer, Fío, pero si no, pues me disculpo de antemano porque a este momento pasamos justo a otras prioridades más de muestreo. Pero voy a decirles acá a los colegas a ver si podemos obtener una vista más cercana de ese quitón.
Oh, my afternoon.
I mean,
Oh, well, it's her shift
Yeah.
at 6. Yeah. So,
yeah. No. So I was from 5 to 4 to 5. And then Oda, I took over Oda because he didn't want to talk.
okay, I see. Yes, I'm gonna go eat lunch, dinner right now, and then I'll be at
Okay,
6.
cool. All right. Enjoy.
I don't know what time it is.
Yeah. Nada más como una visita y cercana.
We'll make sure which one she wants to take. Either those or the ones up higher.
So we're currently gonna towards the end of our dive. Just to remind you that this is dive number 9 of this expedition that is called Octopus Odyssey. We're currently at almost 3,000 meters depth offshore the Pacific coast of Costa Rica. And we have been exploring many different places through our dives. But in this particular dive, we have been focusing on just this spot that is called Dorado Outcrop, in which we find this octopus and many other animals, as you can see on your screen, gathering and thriving around these warm waters that we also call shimmering waters. This shimmering is because of the density difference between the water that is coming out of the crust and the water that is already in the surrounding. And we have a particular question on why some of the arms are so animated, or why they're moving their arms sometimes in a very funky way. And this is a type of behavior that has been proven to be used to ventilate and clean the surroundings around their eggs. So let's remember that the octopus that you see here in this pretty particular position with their suckers exposed, they are the majority females and they're brooding their eggs. So for them, it's really important to keep the conditions around their eggs as optimum as possible. So this is why we see this pretty interesting dancing of the arms. So we have our octocoral expert, Doctora Odalisca Bridi from the University of Costa Rica. And she's very interested in getting samples of these octocorals that are also associated with the shimmering waters. So we will be getting some of these samples throughout the next hour. And just to tell you, and this is a very convenient moment to remind you that all the samples, everything that we have been doing in this expedition has gone under strict permitting processes with the Costa Rican authorities. And because of this, and also because here in Costa Rica, we really appreciate biodiversity, we try to have a minimum impact on the biological populations down here. So when we grab a sample, we try to grab a sample that will impact less. So for example, sometimes if we see a big octocoral, we will just try to grab a little branch of the octocoral. If we have a sponge, we will try just to grab a little piece, and so on and so forth. The same with the octopus. We try just to have very, very few specimens. And on top of this, we just wanted to remind you that all of the samples are going to be deposited at the Zoology Museum of Costa Rica, of the Universidad de Costa Rica. And this is really important because in that way, not only us in the future will have access to these samples, but also colleagues from all around the world that might be interested in these samples in advancing science, they can go and have access at the museum. So this is a way for us to ensure that these samples are going to be kept in the best conditions over the time. Y tenemos una pregunta de nuestros colegas costarricenses de por qué no se observan muchísimos peces a estas profundidades. Y es una muy buena pregunta. Y básicamente una posible respuesta a esto, tal vez tiene dos posibles respuestas. La primera es que a pesar de que acá hay una gran diversidad de organismos y también de peces. Sí que, por ejemplo, si uno lo compara con aguas más superficiales, por ejemplo, en arrecifes coralinos, solemos encontrar a veces tal vez no tantísima diversidad en formas, en familias, inclusive en patrones y colores. Entonces, a veces hay muchísimas especies que se ven que ustedes puedan pensar de que son una misma especie, pero realmente son diferentes especies. Entonces, tal vez eso es como la primera parte de la respuesta, que muchas veces esas diferencias son muy sutiles debido a las condiciones que hay en las aguas profundas. Y la segunda parte de esa respuesta es que como estamos bajando, como estamos bajando en un submarino que pues claramente tiene sus motores, tiene un montón de luces, entonces naturalmente pues provoca un disturbio cuando va bajando al fondo marino. Entonces hay unos animales pues que definitivamente pasan por alto la presencia del submarino y que no tienen la capacidad de moverse, por decirlo así, como estos corales que estamos viendo, porque son sésiles. Pero particularmente los peces. Wow. Nice. Ok. Mientras vemos esta maravillosa muestra que acaban de tomar aquí los pilotos del ROV SuBastian, les comentaba que parte de la razón de que no vemos tantos peces es que los peces suelen ser muchísimo más móviles. Entonces, habrá muchísimos de ellos que al ver la presencia del submarino, pues escapan y no se acercan. Entonces, por eso es importante no solamente saber de que con este tipo de tecnologías estamos muy avanzados, pero también, y se está haciendo constantemente en muchas partes del mundo, desarrollar otros métodos de observación del océano, de las aguas profundas, que no sean tan invasivos y que permitan ver a los animales en su comportamiento natural. Entonces, muchísimas gracias por la pregunta. Y, y bueno, pues de verdad que acabamos de ver un muestreo muy, muy, muy bueno en donde pudimos obtener todo este espécimen de este octocoral que la doctora Odalisca Brady está muy, muy interesada. Ahí se fue. Uy, ahí está. Great. Entonces un muestreo exitoso. So, I would like to take advantage of this moment that we just saw this amazing sample to give a special thanks to our ROV team here in the control room, Tyler, J-Rod, and our very own Jimbo that are behind the scenes and make all of this possible. So a special thanks to them and all the Schmidt Ocean Institute team here on board. And we're gonna keep taking some biological samples around this area, particularly of organisms or animals different than octopuses.
That the white stuff is on?
shots that you want. Just I'm taking straight frame grabs, but make sure like we need the picture first on the scale road.
Son las 7, perdón, las 5:53 de la tarde, hora de Costa Rica. Quería aprovechar este momento para enviarle un gran saludo a toda la comunidad de habla hispana que están viendo estos buceos en vivo y también los que estén viendo esto ya de manera grabada en el canal del Schmidt Ocean Institute. Invitarlos a que dejen sus comentarios, sus preguntas en el chat que tenemos habilitado en Facebook y en YouTube. Todo eso que ustedes escriben lo podemos ver acá en vivo y es muy importante para nosotros pues poder generar esta interacción con ustedes. Y inclusive si no tienen ninguna pregunta, si no tienen ninguna duda, pues solamente déjenos saber desde dónde nos sintonizan, desde dónde se conectan. Y por acá los estaremos leyendo. Y definitivamente los operadores de este submarino son sumamente calificados y expertos. Y como bien lo dice nuestro compañero en el canal de YouTube, con un pulso de cirujano. Así que pues es una gran oportunidad para nosotros contar con este equipo tan capacitado para este tipo de expediciones. Así que, bueno, para mí ha sido un placer, ahora sí, haberlos acompañado durante estas últimas 2 horas en este buceo. Mi nombre es Sergio Cambronero, funcionario de la Universidad Nacional, y los estaré pasando el control a nuestra líder científica, co-líder científica, Beth Orcutt, doctora Beth Orcutt del Laboratorio para Ciencias Oceánicas Bigelow. Beth las va a estar acompañando durante la última parte de este buceo. Así que, de nuevo, un gran placer. Espero que hayan disfrutado de mi compañía. Y yo disfruté definitivamente muchísimo estar compartiendo con ustedes. Así que, pura vida. So, I would like to hand over the dive lead to our co-chief scientist, Dr. Beth Orcutt from the Bigelow Ocean Institute, Laboratory for Ocean Sciences. And we'll also thank to everyone that joined me during this last 2 hours. We have been getting some awesome footage, and also to invite you to stay tuned because we will be seeing more amazing creatures. So thank you, everyone, and pura vida. Beth, thank you.
It's warm over here today.
Yeah, it's just like one pilot, I can get you a compass like that, but I don't think I'm gonna be able to straight up the whole thing. I think that shrimp is done. I'm going for some more of it and put out or just actually put them on.
Riveting. I'll be back. I just don't want to get charged.
Try for some more of it?
We're now. And
we have all the coral quivers available and most of the slurpy collections. Right.
Another one of those.
This one is very good.
Is that yours?
And also, he is more interested in the other one, so I think if we have two.
There's one right here.
instead of-
Want this one?
Yes, another one. Yes, because I think if we have two. Yes. All right, so can we take the highlights while they're doing the questions? So keep track of everything. Yeah, that's fine. I mean, it takes automatic sometimes a minute, but it's just helpful to have more pictures so we can see.
Just got some precision manipulator work happening here. Attempting to sample these very small corals.
Oh, we got it.
Very nice.
Oda, how do you want this recorded? How do you want this recorded? Sorry?
What is this animal?
It's maybe similaria, okay, just octocoral, soft octocoral, so that's soft coral. Okay. That's the corals, soft coral. Okay.
This one's there.
Thank you.
I just took it. Sorry.
Welcome to.
Kira didn't even leave the residue left. Don't pick that thing. Shape this shape.
What do you want next? Okay. You want to?
There are some.
That sponge?
I saw one thing.
Okay.
Maybe we can check.
Okay. Let me just look up here. See if that tells us
We got
anything.
people coming in and out for going to grab dinner right now.
If there's a- I was looking at those
It's a
sponges
nice,
in the back.
a nice sunset outside. Just pop that up real quick. So that's looking off
That
to
might be
the
hard, just because
west
it
off
might
our
be hard to
starboard
land,
side.
But we can try. But would that be your priority, or are there other things you've seen?
No, I prefer to see the
Okay.
corals because around there I saw an Antomasto different to the other one.
Okay.
Sample
I
1 and
see
3,
it
is that
again.
right?
Coral quiver 2.
2, okay.
Just organizing
so is that what we just
the
did?
samples we have taken and
Okay.
anticipating
Okay.
a few more samples.
Yeah, so we're going to come off bottom and have a look to see what the next priority animal sample is. Is it okay if I pan around while you're flying?
Yeah.
And do you want to go up? You want to go over?
Maybe closer.
A little closer?
I cannot see from
Okay.
here.
You can zoom or whatever you want to do. I'll just keep an eye on this picture so I know
Okay.
exactly what I am.
No, it's going to be also
So when we,
Can I see it down here?
yeah,
Oh no, this is the same. We already collected two,
okay,
so we're happy with that.
good.
Maybe you can go over. We already have that sponge, you know. Ya lo tenemos, ¿no?
Stoloniferans, sea pens.
We already have the sequence. Very abundant.
If we could come around to the starboard side a little bit. That one you already have there, this pink one.
This one, we already have this.
Okay, okay,
And
we'll keep
this one.
looking.
Yes.
Coming up or?
Yeah, just up and starboard for now. When we sat down originally, we came from the starboard side and there were a bunch of big octocorals there.
Yeah, this is the same that we couldn't collect. Maybe here.
You wanna
would be
try that?
easier. Yes.
Okay, let's try to land to get some of these white edges.
Edges, yes.
Yeah,
Maybe with a piece of, I don't know. Yes.
We squid up that canal land and it's
With the piece
real
of the
thick
rock,
right
maybe.
here.
It would
Yep.
be-
Yeah.
Not much places to land, I don't know what you try. Maybe
I think I'm lined up.
We tried to collect it a bit before and it was not
Yeah, I was
possible.
kind of thinking the top on that left side where it's a little lower. And then you may have to rotate a little bit, but yeah, I see it.
There's an Antonasu, but there is one that is white instead of red.
Okay,
Maybe we see it. For now, just try to start different.
okay. Tim, if you can get some highlights as we're getting closer, please. Looks like there's a little bit of venting wafting towards us, or maybe that's just the thruster wash. So for anybody that's tuned in recently, we're on the Dorado Outcrop in the Pacific Ocean offshore Costa Rica. This is a site of low temperature hydrothermal venting where waters that are warm to about 12 degrees Celsius slowly weep out of the seafloor here. And we're trying to understand the biology of this environment. Here's the lasers. They're now on the Venus face sponge just above it. Do you see it? Okay, so do you want to try the right side or the left side?
The left side.
Okay,
But with a close up first.
yeah. Okay, so
Probably there would be easiest to grab.
yep.
Maybe, yeah, anywhere there.
I'm just going to put the lasers down a little bit so we can see. Okay, Tim, a highlight, please. Or frame grab, whatever they're called. And then lasers off, please. And another highlight, please, Tim. Okay.
It's like a map. So like
Yeah.
you start feeling
It's very strange.
Another highlight, please. And these are called stolen. Can I write that down?
Yeah, these ones are going to be a tough one for color. Oh, looks cool though.
That's about as close as we can get. Is that okay? Okay. All right. So we got our zooms, glamour shots, and I think we're going to try to slurp this.
No, I think that is better with the
You think it's better with a manipulator? Okay, we'll try that. I guess we need to open a coral quiver first.
number 3 is the next.
So the target animals we're going after are a type of octocoral. Another soft octocoral.
We are currently 2,988 meters below the Pacific Ocean.
Got another, let's say, a little less than an hour to go on this dive. Our intention for tomorrow is to visit
That
a
looks
site
like it peels.
that was discovered just a few days ago. Oh, this is lovely. You see all the polyps closing. Can you take a frame grab, please, Tim? This is a site we discovered a few days ago that is somewhat similar to the site we're at today, the Dorado Outcrop. So we're going to be going back there to try to study the fluid venting some more and see the octopus behavior and try to get some additional experiments deployed. I've never seen a mat like this.
Yes, it's very interesting.
Yeah, our scientists are very interested in this mat-like octocoral. And obviously it peels off the rocks somewhat easily. It's very strange.
It's very strange. Let's stay there.
Yeah, yeah, yeah.
So these are a type of octocoral called stoloniferan. We're not exactly sure which ones these are. We haven't necessarily seen this type of morphology before. Mm-hmm. Ora, you can see like there's some very little ones. Now you can see them there too.
Some fine manipulation from our pilots. No.
Okay. Because I see that this size.
We were just popped free of where we were resting there, so the pilots are gonna reline us up.
Peel, then maybe I'll tear a piece. Sure. That's what I'm trying.
Like waxing.
Pulling up a Band-Aid.
Paraffin coral.
So some of these sea pens in the background that are in that pale pinkish color, we collected specimens like that on an earlier dive.
That's weird.
Is it just gonna keep going?
Yes, keep going. You will see when it's yes. I started very greedy. It's okay.
My fault.
No, I think that I'm very greedy.
Yeah, frequently when our pilots are collecting a sample of coral, if it's something we aren't too familiar with, they will try to get the base material, sort of where it's anchored down to the substrate, as that can actually assist in the identification of many of these organisms. With this, it's just kind of strange. I don't know that anyone in here expected it to peel off like that.
Mm-mm.
But certainly got a good sample with the base material.
You might. Did J-Rod give you a time for off bottom?
I think it was meant to be roughly in the spot.
No, off bottom.
All of them. I'm not sure.
Okay.
To be honest. So that was on deck for 9.
Yeah.
Yeah.
What's your ascent rate? Usually 25 meters a minute?
Yeah, I'm not sure, to be honest. What's the winch speed? Joe, do you know? 0.4. 0.4.
Meters a second? Is that the... Yeah, that's about 25 meters a minute.
Yeah.
Okay, so we probably need to come off bottom by 7,
Yeah.
if not a little bit earlier.
Yeah. Maybe.
So we have about 25 more minutes. Yeah?
Yeah, There is also a little bit of a slowdown for floats.
Yeah, right.
Shallow.
Okay, so maybe 6:45. Okay, yeah, so about 20 minutes.
Fly
Yeah, we will.
a little bit. Did you see some sponges that is just like lollipop?
Yeah, the bigger ones,
No, the small ones.
like these little tiny balls.
Yes, yes. Maybe you can collect one of those.
Okay, that would be your priority, as opposed to looking
yes,
around.
because in this side because I was looking for the Antomasto that was
Sounds like we've got about a half hour left on the bottom here, maybe. And so scientists currently discussing what last few samples we would like to collect from this area.
They're so tiny. I just don't know that we'll actually be able to pick them up. These here, because they look so small. Front and center, right there. That one? Oh, wait. No, she wants these little lollipop things. And so I'm trying to look for any that have a slightly taller stalk. So we can actually get under it. I think that's going to be very hard. Yeah. Yeah. Do you think you can reach up there, or do we need a reposition? Okay. I forgot. Trying to get the basket into view so you can see.
I didn't have to tell people 6:45.
Yep. Thanks.
Good job, Jill. That was awesome.
Thank you.
Mm-hmm. Yeah.
Sounds like we're gonna try and get a sample of these little puffball lollipop sort of structures around.
Mm-hmm. Which
I have a line in 3. Have you used?
the line was an accident. I saw Tyler be like, "Oops."
Okay.
So 3 is open. Thanks for checking.
Which one exactly are we going for?
We're trying for these little white puff balls, so I don't know if that's too far to reach. So there's probably some above us too.
I think I can do that. It's gonna be not the best for a camera view, but I can try. You can try and shuffle over a little bit more.
Before you start, can we get some zooms? Sorry. And lasers on for a moment, please.
There's a big one there.
It's a giant.
Massive.
Yeah, it's probably like that tall. Okay, well, there's a nice little snail there. Yeah, there's the lasers. I'm gonna.
If it works properly, couple of the sponges.
Yeah, we'll try to get a couple of them. We'll just try to suction along.
Yeah, that's,
I don't know if you can actually get all the way up there with this slurp, but
and we could reposition before the start.
yeah, there's also
Just so
some
we
over
can
here
get
too.
with that and get these ones back here.
Yep, we'll start with that.
Okay, I'll, go for this.
Oh, here's two little shrimp swimming by, attached to one another, I think. Maybe it's just one.
Are we second? Second position. Yeah, well, uniforms on. Okay.
Okay, there's one. Okay, I see it come into the suction chamber.
First a wax and now a shave.
Okay, number two. Yeah, they're all going in the same section except for three. No, everything that goes in here is just one sample. And you can mark like how many we end up getting. So far we have three.
Nice.
And four. Okay, I think that's good.
Yeah. Last one.
There he is.
There you go. Switching off. Index. Great.
All right, and now we're going to look for one more thing, which is a white version of that.
Yes, that. Hard to see what number that.
That was number three.
That was number three. but
Oh,
I think
is
it's
it?
back to two. Yeah, it's two. Yeah, I think
Did you just rotate it?
I just rotated.
Okay,
one,
yeah, so
but I
that's
couldn't
number
see.
two, and that's number one.
Yeah.
Yep.
So anything else here?
Not right here, no. So we're going to try to fly around and see a white version of this. That's going to be our next target. So we're getting some larger corals and sponges off to our starboard side. I'm going to zoom in a little bit just to have a look around while you're flying. So we've got a Venus face sponge and a, I believe, a farreid, different octocoral. Yeah. I'm looking for this white Stamathus. Yeah, we can keep coming starboard. Very beautiful large coral coming off to our starboard side.
Yeah.
These corals can grow very, very slowly, so this is Probably a very old coral that we're passing over. Is it possible to get the lights on or the gain up a little bit?
I can just be aware you're up to the right.
Oh, that's true. That's true. Yep. Thanks for that reminder, Jill. Seems to be more sedimented over here where we're going. Nope. There's a unhappy looking fish. Yeah, so we saw those dive weights when we came. Can we come up a little bit towards those dive weights?
Oh, are those Alvin plates?
Yeah, so those
Okay.
are, I don't know that they're Alvin. I think they're actually from a lander because there's so
Okay.
many of them. I don't think that they would use that much weight at once for the vehicles.
Quite the place to land a lander.
Yeah. I think we landed it in the sediment and then carried it up here.
Ah.
Okay. If we keep coming around to the starboard side a little bit, I think I saw something that might be what we're looking for, but I'm not sure. Oh no, that's something else. Okay.
For anyone just joining us, we're currently gliding over Dorado Outcrop at a depth of 2,983 meters below the surface of the Pacific Ocean off Costa Rica. And you're watching live footage from the remotely operated vehicle SuBastian. We are looking around for perhaps one last sample before we leave bottom here. So come explore with us as we cruise along this outcrop.
Where are you?
Wow. Oh, that's too overlapped. Okay.
Still impressive.
I know, but I was like, my God. That's it. Instead of one extraordinarily impressive coral, it's two very, very impressive corals. So
Those are the bubblegum corals. Oh. Yeah, I guess if we can pivot to the port side, follow this. Is there much current down here? It seems that there's a lot of particulates in the water.
Too bad, to be honest. Just for that way where
Jorge, are you seeing anything of interest? One of these little sponges, lacy sponges. Okay, yeah, we can do that. All right, Mike, if we could sit down so we can try to collect this sponge. And I'll get your camera lined up. I think that's pretty close to. Say it again? Yeah, it's still there. No. You all settled in there? Okay. Okay. All right, we've got lasers on. Let me just get them lined up. Can you get a highlight frame grab? And then lasers off, please. Mm-hmm. Yeah. Another highlight, please, Tim, with the lasers off. And then one more. And I'm going to try to get this crinoid. Whoop, too much. Go down ever so gently. Good enough. One more time. Okay.
Could I just get a quick shot of this right here?
The bryozoans?
Yeah, they're just real nice little pattern.
Yeah, one moment.
Go break shot.
Okay. Highlight, please, Tim. At the dandelion.
Oh, there's a dandelion.
Okay. That's a sea dandelion. It's a type of Fauna.
That's what that is.
Yep.
I've seen that before.
And one more, Tim. I will go on closer up.
I can see.
Yeah. Okay. So I'm guessing manipulator into a coral quiver is the best option for this. Do you think we can get it at the base to get the whole cup? Or do you think it's... We can also put it in a bio box. We have almost all of the back is open.
It'd probably be easier to in a bio box if you want the whole thing. You put a jaw there next to it to see for size reference. Yeah, I think you're right. I think it is bigger than it looks.
You're right. Yeah, it's probably that big. Yeah, definitely a bio box. But there's several in this area, so I think trying to get the whole specimen would be worthwhile.
Okay.
So this is not something we've collected yet.
Run them in a little watch out. Don't know if I got the days.
It's okay.
It's free though. I don't have it correct. Wow, there's just nothing.
Yeah, these lacy sponges are very fragile.
You have to get that rocky bits in the back too. So the back bio boxes are all available, are they? Yeah.
Correct.
Escape. When you open up the bow box, go really slow. If you open up too fast, it'll water.
Which one do you want? You've got either go for 2.
Yeah, 2 is fine. It's a large space.
That'll be all right. You'll be good.
So
It's happened numerous times. Like last. You're not wrong.
all right, ready.
You guys still in the back of the
Yep.
box there, chilling.
Yeah, so we've got 2 free riders. We'll see if they stay with us.
So I was thinking if, when we do come up,
Yeah, we have plenty from the site.
great.
Oh, yeah, the little dandelion. Yeah.
So we've had a really great and successful dive here at Dorado Outcrop today. This is the end of our sampling for now. We're going to start heading off bottom to come back and bring all of these samples back to the scientist on board. We expect to have another dive tomorrow, starting at 5 a.m. Costa Rica time is when we'll get in the water. We'll probably be down on the seafloor around 7 a.m. Costa Rica time, which is UTC -6, depending on where you are in your time zone, to think about when you might want to tune in.
I think it's two.
Yeah, we're in number 2 right now.
You can wait till you start. You can just pull your ram in.
This might be the last dive we do at Dorado Outcrop on this expedition. So for those of you that have been joining with us for a few dives so far, we hope you've enjoyed seeing this part of the seafloor. Really a spectacular feature here to have these octopus gardens associated with low temperature hydrothermal venting. Yes, of course.
Bueno, nos estamos despidiendo por hoy de aquí del afloramiento dorado. Ha sido espectacular esta visita a este sitio donde están los pulpos, pero además una gran cantidad de organismos, y ya vamos a empezar el ascenso. Son un par de horas para llegar hasta la superficie, y ahí empieza el trabajo nuestro de sacar esas muestras, procesarlas, fotografías, muestras para genética y preservarlas. Mañana empezamos a las 5 de la mañana baja, como a las hora de Costa Rica, como a eso de las 7 de la mañana. De nuevo, hora de Costa Rica, vamos a llegar al fondo, es el UTC -6 en Costa Rica. Entonces los esperamos y vamos a ascender, y siguen filmando, a veces nos topamos organismos espectaculares en la columna de agua. Les habló la doctora Beth Orcutt anteriormente, y ahora Jorge Cortés de la Universidad de Costa Rica, y nos vemos mañana.